Pilot, we'll wait for your confirmation of thrust before releasing the vehicle.
Pilot understands. Pilot does not have control. Stand by. No control.
All stations, we're recovering.
Bouncing box. Dive Super Pilot, the control box has been cycled. I don't want to test the thrusters until I have your confirmation, so we're standing by.
Good copy. We're holding in a good spot. You should be clear to test thrusters.
Copy that, testing thrusters. I have thruster control.
Good copy.
Confirm.
Are you ready to redeploy?
Control room ready? We're ready.
Good copy. Let's we'll hold off for a second as we get our streaming back, and then we'll put it back in the water.
Understood.
All right, all stations, we're going to continue with the launch of the vehicle. We're going to go ahead and lower it down to the water now. Pilot, we'll wait for your confirmation of thrust.
Roger. Pilot has good control.
Pull the pin. Lift line's clear, pilot. Go ahead and lock in auto heading, inverse the ship, and push out.
Pushing out. Full
Thank you.
steam. Yep.
Appreciate it.
Auto heading engaged. Full thrust.
That's a good heading there.
Roger.
It's looking good, pilot. We can go ahead and go off stick. Leave auto heading engaged.
Pilot off stick. Auto heading engaged.
All right, deck. Let's go ahead and launch Sirius. Pilot, please provide some halfway as Sirius comes up and over the side.
Understood. Backing. 30%.
Copy.
Increasing to 40. Backing 50.
Good copy, pilot. I'll have you increase back.
Increasing. 60.
It's looking good, pilot. Let's maintain. We're bringing it center line.
Roger, maintaining. Easing.
Go ahead and ease, pilot.
Easing. Pushing out.
Good copy. Winch 0 reps.
Zeroed.
Pilot 0 turns when you're aligned.
In work. Turn 0.
Okay. Pilot, let's go ahead and descend down to 15 meters per minute. We'll call it every 10.
Roger that. Descending 15 meters per minute.
Descending.
Ready for video.
One 0 meters, wire out.
D2, 15 meters.
Two 0 meters, wire out.
D2, 23 meters. All's well. Pushing up. Yep.
Three 0 meters, wire out.
D2, 33 meters.
Dive Super Nav, we have good tracking.
Good copy, Nav. Four 0 meters, wire out.
D2, 43 meters.
Five 0 meters, wire out.
D2 holding 5-2 meters depth.
Winch, dive super.
Go ahead.
Can we pass control to the control station, please?
Control room has control.
Control room confirms winch control.
Pilot, dive super.
Go ahead, dive super.
If the control room is ready to continue with the dive, please do so.
Understood. Thank you, dive super.
Nav, dive super.
Dive Super Nav.
How's our approach to bottom?
Yeah, we went considerably further west than planned, probably mostly due to the current swinging around more westerly. So I think if you could get them to ease into auto position before you hand off, that'd be helpful.
Will do. We also have a slight heading change to do so, most likely. So be advised, we might be coming to port up to 20 degrees.
Good copy. Standing by.
All right, we'll come into auto back here. Nav, super.
Dive super nav, go.
We're in full auto pause, passing the bridge at this time.
Understood.
Nav, dive super.
Dive super nav.
Bridge has good control of the ship. Auto surge and sway are engaged. The heading is now a 090. Center rotation is midships. Port prop and rudder are still disengaged at this time. I'm ready to pass comms to you if you're ready to take.
Nav copies all. I am ready to take comms.
Nav has comms.
Nav has comms. Thank you, dive super.
Go ahead, Nav. That's correct. We have port prop and port rudder still disabled at this time. Alright, sounds good. I'm ready to start position move.
Okay, we have a long way to go, and
Go ahead. Sounds good. We'll start the position move. Range 730 meters, bearing 097 degrees. Speed decimal 2 to begin with, and slowly increase if possible. And do you want us to notify you if we couldn't make to decimal 5?
yes, please.
Alright, sounds good. Thanks.
Headset test. Headset test.
Nav bridge. So we're doing a position move right now. The ship holds decimal 5 pretty well because it's a heading that it's like the similar direction as our heading, and we're just using our prop to move forward. And we can even increase that speed if you all want. Would you like that?
Decimal 5 is good. I don't want to get too much cable layback. Decimal 5 will get us to our waypoint with sufficient time, so let's hold decimal 5.
Alright, sounds good. We're at decimal 5 right now, and if at any point you all want to increase speed, just give us a call.
Okay.
Thanks.
Good copy. Thank you, Bridge.
Pilot, I think your winch next. I just checked it and it's looking good.
Copy, thanks, Winch.
Winch, this is video. How's your side tone?
Side tone check. Side tone check. I think it's good.
Copy.
I did yank on this cord the other day. Pretty hard. Maybe it's intermittent connection, but side tone seems good now. Okay. Going off comms.
Copy. This is Okeanos video engineer Roland Bryan making a general announcement to all those on the ship and the shore. This is a reminder that all audio associated with this dive will be recorded with our archive video, as well as being streamed ashore via the satellite and the internet. Keep that in mind as we proceed with the dive today, and enjoy the dive. Thank you.
All right, good morning, everybody. So after a couple days of transiting through the Gulf, getting bounced around a little bit, looking for good weather, we are approaching bottom on our 15th dive of this expedition. So we've left, our last dive was the Seep, which was pretty incredible. I think having a couple days to digest everything that we looked at was useful. But we've kind of gone across the entirety of the Gulf, and now we're, I believe, about 20 miles south of Middleton Island, which is a small island in the northern Gulf, diving on a place that we're calling the Middleton Canyon. We're going down to a depth of about 2,200 meters and planning on climbing up one of the ridge lines along this canyon. And so I don't think we know too much about this other than some bycatch records of corals and sponges, so it'll be exciting to see what we see down there. And with that, I'll pass it on to the science leads to introduce themselves.
All right. Thanks, Sam. And good morning, everybody. My name is Jamie Conrad. I'm the geology lead on this expedition. I'm a geologist with the U.S. Geological Survey. I work at the Pacific Coastal and Marine Science Center office in Santa Cruz, California. So Sam mentioned, today's dive is in a canyon system offshore southern Alaska. We're about 100 kilometers offshore Prince William Sound, and in a canyon area about 2,200 meters depth. We're technically, geologically, within the Aleutian Subduction Zone, but this is a very different area and subduction zone than we see extending for about 3,000 meters to the west. Instead, there's a large crustal block known as the Yakutat Terrane that is impinging on this part of the subduction zone. And the Yakutat Terrane is a crustal block that originated off the coast of Oregon and Washington and was detached probably around 30 million years ago and transported north on the Pacific Plate at about 2.5 to 3 inches a year, 55 millimeters to 60 millimeters per year, to where we see it today. It forms a large block that extends from where we are to the southeast, offshore, southeastern Alaska, about a midpoint of the Alaskan Panhandle, about 500 kilometers, and forms a broad shelf about 100 kilometers wide off that part of the northern end of southeast Alaska. Part of the block is actually being subducted and extends actually for several hundred kilometers underneath North America in this little corner of Alaska. But the upper parts of that block are getting scraped off into the accretionary complex and once again becoming part of North America. The particular area we're diving in, I expect pretty complex geology reflecting that collision. Lots of faults and folds, deformed rocks. We'll see just a small window of that, but whatever we see, any rocks we can collect will be very useful for chemistry, and any observations we can make will be very valuable. I know I say this before every dive, but I really mean it this time. I really have no idea what to expect here, so should be very interesting. And with that, I'll pass it along to our biology lead.
Hello, everyone. I'm Merlin Best. My pronouns are they, them, and I'm a biologist with Fisheries and Oceans Canada. I work out of the Institute of Ocean Sciences in Sydney, BC on Vancouver Island. Excuse me. So yeah, the general area we're in today has been marked as one of interest by Sean Rooney at the Alaska Fisheries Center. He, I believe, works with data from various trawl and longline surveys, which has historically been the main way that we get information about habitat from the bycatch that we encounter on those surveys. Those fishing techniques can't be deployed in rocky habitat, so trawls and long lines, and they're also limited in the depths that they can go to, usually 700 meters max, sometimes as much as 1,000 meters. That means that anything rocky and anything over 1,000 meters is largely unknown across the whole shelf in this region. So this is an excellent opportunity for us to use our platform, the ROV D2 or Deep Discoverer 2. We can take it down and look in a place that's kind of a blank spot on the map for biology. Whatever we find down there, even if there isn't all that much to see, would be an important data point for scientists like Sean and others to use for their analyses. We'll be documenting things like the identification of the animals that we see and their abundances, so how many of them we see. We'll likely be landing, I think, on softer sediment, so it could be a bit more sparse and get into more complex habitats if we get into more of the rocky substrate that we're looking for, where corals and sponges like to grow. And yeah, we mentioned the records that we do have, so from generally shallower than 700 meters, indicate a couple of interesting finds nearby, some Gorgonian corals, these larger structure and habitat forming corals, and also a couple of glass sponges, the cloud sponge, Afrocallistes vastus, which is one of the main species found in sponge reefs off of BC. So a couple of really interesting points nearby. We'll just have to see what emerges from the depths when we get there.
Yeah, I think it'll be interesting to kind of compare what we see here to what we saw in Noyes Canyon. We're a bit deeper here, and it's kind of a little bit of a different terrain, and we're across the entirety of the Gulf. But it'll be interesting to see if we see kind of similar distributions, different. I know in Noyes we saw a lot of things pointing to it being a somewhat nursery, so it'd be cool to see if we see similar things here.
Yeah, we found those two octopus, deep sea octopus, and a bunch of juveniles around, so an active octopus nursery. You never know what you're going to find in these canyons. And we're on bottom, I think.
Yeah, just coming into view.
Look at that for timing.
Nailing it. Ooh, that fish down there at the bottom is a Antimora, the Pacific flatnose. You can tell it has a really distinct tail. And I think we saw a pretty large grenadier or rat-tail fish on the way down. And that right up ahead looks like a flytrap anemone. So in the family Hormathidae.
Which is how we started our Noyes Canyon dive.
Ooh, that's true, yeah.
Wow.
That's right. These anemones were kind of lumped on every single rock that was available. I think just below that flytrap anemone, there's another kind of a white base with purplish tentacles, a different kind of anemone. I think I had a tentative idea of something like Actinernus nobilis is a pretty distinct looking species.
So we see right off the bat a fairly soft, muddy bottom. And perhaps another comparison to Noyes Canyon a couple days ago, which was just a few miles away from the very active and fast-moving Queen Charlotte Fault that shakes the area pretty hard. Here we're kind of on an area that's under tectonics on steroids. We're right over the rupture area for the Alaska 1964 earthquake. That was a magnitude 9.2 earthquake, one of the, I think, 3 largest that's ever been recorded. So you can imagine that we're in an area that sees very strong shaking from time to time. And so we might expect to see some evidence of that, debris flows, slope failures as we head up the side of this flank of this canyon. And one thing that kind of struck me looking at the backscatter and bathymetry data today was the canyon bottom. We typically think of these canyons as being inactive, these that are this far from land. They're kind of cut off at this point from sediment sources, and so at high sea level stands become inactive. But to the extent that this area is, that the canyon does have anything moving down it, it would likely be related to sediments that have been shaken from the canyon walls. So we'll be looking for that.
Beautiful zoom on this anemone here. Looks like there are several different species right in the area that we landed, and while I don't know many of the different kinds, we can document with these really excellent zooms and go over the video later with experts to see if we can separate them into morphotypes and maybe figure out what species they are. Ooh, wow. We've got a nice close-up of the mouth, and you can see surrounded by all of those tentacles. If we end up seeing corals a little bit later, you can see how they're related. They kind of have the same basic structure, just generally shrunk into really small polyps attached to a larger skeleton. That's great. And maybe just a quick zoom on the crab to the right for ID. We can make our way forward. Oh, excellent. Thanks for the reminder. ROV watch lead. I'd also like to request a Niskin maybe after this close-up.
ID EX2306-D15-01W at Niskin Bottle 1.
So this is looking to me like one of the species of tanner crabs found in this area. Not my area of expertise, so we'll definitely capture this really nice imagery, and experts can look over to confirm and maybe get it down to species. They'll look at things like the ornamentation on the shell, all those little spikes, kind of the overall shapes and distributions of them.
Looks like he's been digging around in the mud. He's covered with that kind of greenish looking.
Yeah, it does. We're maybe kicking up a little bit more. So the science chat is looking a little quieter than usual. It is a Sunday. While we're diving today, we have a science chat open. So it's our chat room that's populated to varying degrees by our shore-based scientists, who are scientists working from all over the world and all over the country to pipe in with their observations and IDs, but don't think there's anyone that can help me out with crab ID today. That's okay. We have a grenadier up on the right, and some more of those flytrap anemones, and maybe a different species, this flatter one. It's hard to say. That'd be great, thanks. I thought it might be a sea star arm, but it looks like something else. Yeah, dead piece of something for sure. Yeah, it looks like it might have been attached to a rock or something at one point. Oh, I see. Does look like the remains of possibly a coral skeleton next to this anemone.
Still attached to the rock, looks like?
Looks like it, yeah. So I'd imagine it would have been on top. Corals like to grow up. And so it looks like this rock has maybe fallen over. That looks like a bamboo coral skeleton to me. You could even get a tentative ID. You can see that it's branching right at the nodes, I believe. But almost certainly a bamboo coral of some kind, unless that what looks like banding is actually just kind of detritus. So it could be something else. Ooh. Yeah, I think I'm seeing a really large, darker band in between the white skeleton. That tells me it's a bamboo coral. And I think those are little brittle star arms gently waving from underneath of this rock. Little crustacean on it, maybe a Munopsid. That's great, thank you.
So he's still attached, but sideways, suggesting that the rock has tipped 90 degrees at least, maybe part of his demise.
Yeah, I think that would do it. If it completely fell over, and then especially if it came any distance down this little hill. it's getting quite steep here. Oh, there's some rock. Ooh, and a large brisingid. So kind of a type of sea star, but they're in their own order. They generally stay attached to rocks and reach out into the water column or around to search for food. Several different types of anemones, and 2 different kinds of black corals as well, just in this area. And those white things in the back are the carnivorous sponge, Cladorhiza cornata. Excellent.
All sitting on a very, very beautiful example of conglomerate, pebble to cobble conglomerate, probably deposited here as part of a debris flow down this canyon. And just a little bit of everything in this deposit. I see granites, metavolcanics, probably fine grained sedimentary rocks, siltstones or metasiltstones and mudstones, just with a quick glance.
Wow, and I'm seeing some amazing detail in this zoom of the brisingid, this the central disc covered in spines, the attachment point to the individual arms. These are all things that can help experts assign an identification to this species. Yeah, really big flytrap anemone beside it. I think I saw a different kind of carnivorous sponge just below, and then in the back there, some of the, it's a very cool carnivorous sponge. It has a little kind of crown on the inside where it gets its species name. So you've kicked up a little bit of sediment. I think it's probably going to be something we struggle with for the first little bit of this dive. That's okay, you can just kind of slowly pan over what we see and get out of this cloud. Over to the right, that's a black coral, so it has a dark black skeleton covered in this lighter orangey colored tissue. And yeah, those very distinct, they kind of look like lollipops almost, white stalked carnivorous sponges.
Not getting much current here to help sweep away the mud we've kicked up.
Mm-hmm. So we're getting a nice zoom of this black coral. It looks like there are parts of the skeleton that are dead. They're lacking the tissue, but other things are growing on it. I think those are amphipod tubes. The seem to make little dwellings for themselves out of mud and other kinds of sediment. You can see their little, little parts of their arms, I guess, sticking out in the parts of that coral that have no tissue. Could we get a zoom on, I think you might be setting up for one, the white-stalked sponge? Ooh, it actually looks a bit different now that we're getting closer. Wow. So this looks the most like a Cloudorhiza corona, but it's very different from the ones that I've seen pictures of, where it has a second kind of, crown-shaped, I guess like a head on the stalk of this sponge. It's a very different morphology from the ones that we've seen. I wonder if as they get older, these might be older specimens that have kind of extended the features that we see on younger ones. Also the little, those 3 rays coming out from the middle of that disc are really long compared to the ones that I've seen. This is very important imagery to collect, if that's the case, unless we're looking at a different but related species. So at the end of those thin rays, actually running all along them, I believe this species has little hook-like skeletal components that will act kind of like Velcro and catch and tangle up really small invertebrates, probably crustaceans, little zooplankton that land on them. They get caught in those little spicules, and then eventually the sponge will grow over them, and they'll get absorbed into the tissue. Ooh, actually, yeah, the barnacle, please. Wow, this is really cool. So just starting up the base of this ridge that we're climbing, this is a stalked barnacle. Oop, it just retracted its little feeding appendages tightly into the, the hard part. But you can see, wow, you can see it really move. So I think this is probably a scalpellid barnacle. A number of different species of stalked barnacles are found in the deep sea. That's great. If we could get I don't know if we got the lasers around it or not, but yeah, just for a quick reference, that's great. Thanks. Sure, yeah. Yeah, it's cleared up a little bit, so I think it's definitely worthwhile.
Actually, just confirming that coral on the left, is that a different?
It is, yeah, it's a different coral to the one that we looked at for sure. It's quite a bit to look at, just in this one small rocky area. Okay, so an interesting kind of elongated shell on this gastropod, a snail or a whelk of some kind. It seems kind of longer and smoother, not as many ridges as some of the species that we've seen so far. I think it has a little, at least the beginnings of maybe an amphipod tube on the shell. That's great, and maybe, yeah, that's perfect. And yeah, if we could quickly snap on the coral to the left as well, then that should be one of everything in this area. So if we get a nice close zoom on, every new thing that we see, it's a lot of work in the beginning, but then we can kind of travel over things confident that we've collected good imagery enough to potentially identify it as we go forward up this feature. Yeah, definitely a different black coral. Really different branching pattern from the bathypathes that we saw earlier. This one has these four branches coming out kind of at the top that are in turn branching themselves, so a bit more complexity. You can see the dark black skeleton showing through the almost translucent tissue. That's great. So those two red laser dots that you saw are exactly 10 centimeters apart. So we use those as we're exploring down here to kind of get a sense of the scale of what we're seeing. There's a sea cucumber. We've seen those kind of before. We can just maybe take our time moving over this rock. I'd say that black coral's even different from the one that we just saw. So that's 3 different species of black coral so far. Yeah, take your pick. There's a few. And this one again, branching pattern is the main way that we at least roughly decide which group the black coral that we're looking at is in. And this one again, a distinct branching pattern from the 2 that we just saw. This one could be something more like lilipathes. Oh, and one of those little benthic jellies right at the base. Ticogastria. And again, there's some dead material with those amphipod tubes encrusting on the top. Beautiful. So you can see pairs of individual tentacles coming off of this. So much shorter tentacles and, let's say, smaller polyps than the bathypathes that we looked at in the first, right where we landed. And you can see these smaller little branchlets that we call pinnules are coming off in, I think I'm seeing, well, 2 to 4, but I think overall 4 separate rows, which is one of the ways that we can tell it's likely in the genus Lilipathes. And there's that little rock-associated jelly, the Ticogastria. It's the species Ticogastria polaris that we think we've been seeing quite a bit of. I think the rest of these corals look the same to me. So we can just kind of slowly fly over them and we can just document their abundance.
Yeah, I don't know if that big boulder is part of these conglomerates, but it could be, I guess. These are pretty coarse. It might be a little bit of a puzzle as to how you get big boulders and rocks like this out. We're close to 80 kilometers, 120 kilometers, or perhaps 80 miles offshore. But likely, and I don't know that the glaciers did get out to the edge of the shelf in this particular area, but I suspect they did, looking at this stuff. So these would be boulders and cobbles that would be pushed out as part of the movement of the glaciers out to the edge of the shelf and pushed over the edge, perhaps. They then later would be cascading down the canyon as debris flows or turbidite flows, perhaps triggered by big earthquakes, or maybe even not so big earthquakes.
We're just trying to get a close-up. Obviously, there's a bit of some of the sediment really stirs up anytime you get close, but I think those are juvenile mushroom corals. looking at the size of the polyps and kind of their morphology and the color a little bit, they're, yeah, I think 2 small mushroom corals. Don't know what that thing is to the right of them, though. A really large shrimp of some kind. Bigger colonies of this probably lilipathes coral. Ooh, a really big bathypathes coming into view on the right. There's a glass sponge just above it. This is just a really fine, you know, silt or, you know, maybe some of the accumulated marine snow is really susceptible to dusting up on us. You can see there's just a fine layer of it over most of the rocks here. Ooh, this is a cool fish. This might be one of those, kind of like the snailfish that we saw before. Ooh. So I think we're looking at a snailfish, a leparid or leparidae. And these are fairly rare to see. We've only got video, I think, of one nice close-up like this one. Some of the deepest living fishes that we have on record are snailfishes like that one. You know, the imagery that you see from dives, you know, 7,000 meters depth, the images that we get back, the fish that are in those are generally snailfish.
I'm starting to get the impression, looking at the outcrops on the way up, that these conglomerates are perhaps interbedded in a fairly thick sequence, sedimentary, obviously, sequence with some other lithologies, and you can see that a fair amount of deformation within those more massive rocks that it's hard to pick up in the conglomerates, but suggests that these rocks the conglomerates are actually fairly old. Here you can see a nice contact of possibly some sandstones that are lying above that conglomerate outcrop. So these might be part of the far more ancient cover of the Yakutat Terrane that has become accreted onto the North American continent here.
Wow, really different up here. Some really large, probably lilipathes corals. Lots of those anemones, the purple and white ones that I think are something like Actinernus nobilis. Couple of ceryanthid anemones kind of sticking out of their tubes. And that yellow ball in the upper right is a what the flytrap anemones look like when they're retracted, when they're kind of all balled up. Oh, this coral right up front actually is a bit different. So the big one in the, yeah, right there. Actually, the branching pattern on this one's even different from the ones that we've just seen. So that's, yeah, another species of black coral. Ooh. This one is interesting. If anyone in our science chat, I see there's a few more people that have joined us now. This doesn't quite look like anything that I have in my guide. So this, yeah, I'd love a nice zoom on this one. Maybe Dendrobathypathes has some kind of similar branching, but yeah, very different from what we've seen. Ooh. So you can see a little bit of the dark black skeleton peeking through. Just trying to get focus on the smaller branches and how they're coming off of the main branch. Very interesting. Just pausing to see if anyone in our science chat has any suggestions for the ID of this one, or if it would be worthwhile to take a sample. I think I see a little amphipod, some glowing amphipod eyes sticking out from the branches. Okay, that one's very interesting.
Want to grab a piece of that?
Well, hoping for a vote from the science chat. Just because I don't know what it is doesn't mean it's not known, but it is. ROV, how would, are we set up okay for our collection if I... Okay, I think, yes, yeah, the one that the lasers are on, and it would just be a snip of a branch. Oh, good. I just seeing now that the science chat agrees and would like a collection. So we have that it's possibly a cladopathid black coral, not 100% sure. So we have this excellent in situ imagery, and if we grab just a small piece that we can take back to the lab to give a confident identification on, then we can use that information going forward. When we see more, we don't need to collect it, but we have a very good idea of what species we're looking at. So this will be a really useful reference collection. And Harvey, just a reminder, these skeletons are really tough, so you definitely have to snip it. It's perfect. Mm-hmm. Mm-hmm. Nope, right there is great. Yeah.
That's great.
Yeah, that will work. Yeah.
Ah, nice.
Mm-hmm.
Almost all broken off. I think he's not on the cutters, perhaps.
The last one.
Yeah, these black corals are really wiry. Oh, there we go.
Nice.
Beautiful.
Very nice.
Whew. Beautiful collection. Thank you for that.
ID EX2306-D15-02B in the port inboard bio box.
ROV, when you're ready, I have a Zoom request for this area. So another one of these black corals, I just had a request from the science chat to zoom in on it again. Yeah. So I think there's one to the right. So not the one that we collected, but one nearby. Yeah, that one there, kind of wider branches in the middle of that clump. So just to the right and above, up of the lasers. Yeah, no, not the really bushy one. So actually the one just to the right now, this might be a better angle. The next one over. Any of these, there's a bunch that kind of, so that one, yeah. Yep, that one's great. The science chat has noted this is a real black coral garden. I think that's 4 different species, different genera of black coral that we've seen so far, just in the short time we've been down here. So that's Excellent. Just to get an idea of the branching pattern that we're seeing. Might be able to figure out what species it is. And again, it's got a little bit different of a branching pattern at the top here. The main stem looks like it has, I think, 4 rows of branches coming off of it, I believe. Maybe just the 2. And 4 rows on these smaller branches. That's great, thanks. Can zoom in a little bit more. Beautiful. So you can see those pairs of tentacles, really different morphology from the octocorals that we see. That's great. Thanks for that, RV and video.
So these more massive rocks have the look of being fairly thoroughly or somewhat tectonized and partly metamorphosed, which makes me think they're probably relatively old, perhaps originally deposited off the shore of present-day Oregon and Washington and transported up here. Could also be related to the collision of the Yakutat block with the Aleutian Subduction Zone, or up at this level, the accretion zone. That process began about 10 million years ago, so these could easily have been deposited in a basin as the block approached the subduction zone. Ooh. And ultimately buried and metamorphosed under a fair amount of pressure. But you see a lot of signs of shear and joining and fractures that are running vertically through the rocks. That tells us they've been fairly worked tectonically.
Well, yeah, very interesting. Probably an octocoral holdfast. We saw a bamboo coral one earlier. This could be the same. And also another nice view of, I think, the coral that we collected up above. There were some nice cereanthids for whoever on the science chat was looking for cereanthids. We did manage to collect a couple on a previous dive. And I believe, too, really large examples of the one that we just collected, I think. That's great. Thanks for catching that. I think on that rock at the back there, too, there's a few coral holdfasts. Yes, please. Yeah, I also think it's a sponge. So it's pretty small. Again, those laser points are 10 centimeters apart, so probably a juvenile of something larger, although not necessarily. And very cool geometry. There are a couple of things these really small ball-shaped sponges could be, so it's nice to document. Oh, very cool. Still hard for me to tell what it is, especially when it's this size. Definitely those little crustaceans seem to like it. Someone else in the science chat pointed out you can see little brittle star arms waving from underneath the rocks and in the crevices.
There they are.
I haven't seen any of their discs. They seem to be really wedged in there. Ooh. Wow. Okay, so I think I'm seeing kind of some of the hallmarks of a glass sponge skeleton, a really thin mesh of spicules, those silica-based skeletal elements that make up the skeleton of the sponge. That's great, thanks for that.
See a contact. Wow. Oh, beautiful structure. Can we zoom in on that, the rock that's just underneath the lasers now? You see some very nice cross-bedding.
That's what those like kind of angular patterns are?
Yeah, the laminations. Yeah, kind of, yeah, the big square one kind of right in the middle. You can see the striations or banding on it.
Mm-hmm.
Oh, very nice. Tough to see exactly underneath all the coatings, but we're right on, you can see this was probably a sandstone bed right above those conglomerates, a depositional contact. With the banding, you can see, tells me this is probably a turbidite unit. That's a submarine, kind of a mass flow deposit. They can travel quite far underwater, and probably related to the same sort of mass wasting and debris heading down a channel, submarine channel system that the conglomerates represent. Okay. Pan down and just have, or just back out a little bit and have a look at the contact with the rocks below it. And there you see the conglomerates that were just underneath it as this bed then moved over. But it's interesting, the sandstones seem a little bit rare here. We're getting mostly these really coarse conglomerate that are deposited.
Some really large brasingids. They're, I think an anemone, yep. Maybe zoom in on this sponge, the white kind of folded one coming up to center. Ooh, there's another one down there. So this is a glass sponge in the genus Pharia. It's there are a few different species, kind of known, quite a few from the Gulf of Alaska and farther south in BC in the areas that I study. So it'd be hard to assign a name, species name, but the genus Pharia, some of these are associated with sponge reefs. We often see them in kind of higher numbers on seamounts. You can see parts where the skeleton have kind of died away, or sorry, where the tissue has died off. It makes these really intricate, geometric patterns, this mesh. Everything in this class, Hexactinellida, has a 6-rayed geometry. So when you get those 6-rayed individual spicules interacting with each other or fusing in some cases, then they create these really beautiful patterns. So you can see the, closer up front, the living tissue kind of covering up the skeleton that you can see through in behind. That's kind of a smaller individual, but you can already see there's lots of, shrimp associated with this one. Just a beautiful shot of this brisingid. Yeah, I thought I saw it kind of to the left, I think. No, that one I think we got earlier. That's okay. I think we'll continue forward.
Science, we're just trying to get a good image because the previous one was super dusted.
Sure, of course. Of the sponge? Yeah, okay, great.
There's a brittle star out and about.
Ooh,
making
yeah.
his way up the slope.
Kind of the first one that we've seen exposed. So there's also Cladorhiza bathycranoides, in addition to the Cladorhiza corona, that have this general shape, but I think corona is the only one with this little on the inside of this disc, that little crown. And look, you can see crustaceans kind of enveloped in tissue. You can almost see through in parts.
Now, are they on the backside or are they in it?
I think that they're in it. Often when I get these in the lab, you can cut open some of these little balls, and they're full of, partially digested legs and things like that. That one, the ball at the base of these is a little bit bigger than what I was expecting. I was expecting kind of smaller organisms. I'm not familiar with this exact species besides, what it broadly looks like. But a very beautiful and deadly, if you're zooplankton, carnivorous sponge. Because they're quite small and delicate and white, all of those things make it really difficult to photograph. So we'll try to use that really excellent zoom and that laser measurement to give us an idea of what species it might be. There's a couple more brittle stars. A little crab. Wow, a really steep rock feature underneath of us right now. There's a mushroom coral we haven't seen yet. The big red thing to the left, upper left. Mm-hmm. Yeah, we can just do a quick a snap zoom for ID. If we can get a decent view of the cap, and we can kind of look for the different kinds of polyps. Might be a little difficult, but I do see a smaller size class of polyps in between the really large ones, and maybe a third, so that can give us important information as to the genus of this one. The mushroom corals in the area are in 3 different genera. There are a couple of undescribed species out there, one of which we were able to collect on an earlier dive. And so this one, with the close-up of the cap of the mushroom, will be really helpful. Thanks for that.
And the substrate, you see, is kind of full of holes. I wonder if there's a little pocket of relatively recent mudstones that's clinging to the side of the slope here. But those are just a little one. I see some pretty rocky-looking stuff all around it.
It's another faria with really large kind of folds or almost tubes. I've never encountered mudstone until this this survey.
Well, and these are pretty soft, as you saw. They look like rocks, but they crumble in your hands, or turn to clay.
So we've had a tentative ID on that mushroom coral, possibly more related to Pseudoanthomastus. So the genus Pseudoanthomastus. Seeing more shrimp and more of that lilipathes, the Actinernus, anemone, a couple of cereanthids. Oh, interesting. Is it a sponge or an urchin test? I think it's... Oh, there's a little carnivorous sponge sticking out of the soft substrate as well, I think. And that sponge above, another one of the white ball-shaped sponges. They're very small. Really hard to tell what kind of group they might be in at this size. You can also use these really tight zooms to take a look at everything else that's in the sediment. So most of the animals found here will be the things that we don't see because they're in the sediment. Different kinds of worms and other animals that spend their life, buried in the sediment. Oh, could we zoom on that coral, the long white one? I think this is the first bamboo coral we've seen, at least alive. I think we saw some of the dead skeletons farther down. Just a snap zoom. I think we've seen this one on previous dives, but just to confirm. So these long, unbranched bamboo corals could belong to a few different groups. At one point we called them all Lepidopsis, but now we're going off of things like the sclerite arrangement and the polyps, those hard skeletal elements you can see like that look like little lines at the base of the tentacles. This one's a little bit interesting. It doesn't have a really big spine coming out from in between the tentacles like some of the other ones, so it's a slightly different group than some that we've had really good close-ups of on previous dives. This is excellent imagery. Thanks for that. So I think while we're looking at the taxonomy of these corals, we have in the meantime have placed them into groups, so this one will be assigned a group based on what the polyps look like in the arrangement of those sclerites.
Has the ability to characterize these things by DNA led to some changes in the way these are classified, some of these creatures?
Absolutely, yes. I think, yeah, the more DNA analysis we can use, the more that we realize that certain characters that we've chosen to differentiate them might not be based on the way that they're related, that, you know, branching from the nodes or the inner nodes or not branching at all has kind of arisen several times independently in their evolution. So, so yeah, we've had to move gears from the classical way that taxonomists have differentiated them to reflect what we know now from the genetics. So that's why I can no longer call this unbranched bamboo coral Lepidopsis. It could be closely related. No, I think we're good to continue up. Yeah, in some ways, my life is a little bit easier. You know, it's really easy to tell something like branching pattern. It's a very accessible trait, I think, when you're just starting out. But the more that you look at individual polyps of corals, the more different they look from each other. So there's some things in that one that make it quite distinct from the other long, unbranched bamboo coral we saw. Oh, can we look at this this bushy one to the right? We've also had a sighting of a Panikia-like holothurian on the sediment-dusted rock. Interesting. Okay, so this is actually another mostly dead skeleton that's, I think it's been colonized by, well, oh no, maybe not. It's just kind of looks like it's in trouble. Some of these get colonized by small anemones, kind of colonial anemones called zoanthids, so it makes the texture look really different. But these ones look like bamboo coral polyps that have been damaged in some way. You know, a few of them are kind of look like they're missing some tissue and they're balled up, but a couple of them in behind look healthy and kind of active, and you can see the banding, that dark band on the white skeleton that tells us it's a bamboo coral. So some kind of sparsely branched bamboo coral with some damage. That's great. And a really flattened looking flytrap anemone in behind it.
Oh, is that what that is?
Yeah.
Okay.
That's great. No real indication of what's been causing damage to this coral. It's great. Thank you. Oh, wow. Can we look at this one, please? It is. It's a really nice view with the really dark background. So this looks like a primnoid of some kind, I would guess, from far away, but let's get a little bit of a closer look. So this one is a Peristanella primnoid coral. So smaller polyps, I think, than the ones that we were looking at on the bamboo coral, coming off in kind of these neat little whorls. And then another one of those groups where there are several different species it could be. They're very difficult to differentiate in situ. They're even a little bit hard to do in the lab. So I think we'll stick with the genus Peristanella for now. Very nice. You can see, I think, a brittle star arm intertwined, and definitely a lot more of the polyps kind of facing the other way. So this is kind of, we're seeing some bare skeleton on this side. Definitely a preference, probably based on the way that the current flows through this area. Yeah, that's okay. Yeah, I think we'll... Yeah, I love being able to see the sclerates on these things, but I think we're a little bit far, and we might see another. There's another dead bamboo coral skeleton off to the right, too. There's lots of stuff happening on these rocks.
Well, the rocks themselves are looking pretty beat up. Lots of fractures, vertical, subvertical fractures, and you can see pieces are starting to break away. And I'm not seeing that real conglomeratic look to it. I see maybe something that looks like bedding. It's hard to say.
Ooh, wow. Okay, this zoom is incredible, so we can get an idea of the overall morphology of individual polyps on this primnoid coral, this Peristanella. You can tell, I think, just by looking at them from here, they're really covered in these hard calcium carbonate sclerites. I see them as a bit more armored than the bamboo coral that we saw, so that they're almost stiff, and you can sometimes barely see tentacles sticking out. but this information, the overall shape of these polyps, could help us narrow it down to species.
Looks almost like a snowy slope up above this.
Wow, does it ever? Look at all this. Ooh, there's a really interesting one. Can you go towards that lower right? There's a dead coral skeleton, partially dead, and then whatever is attached to it. This kind of yellowish. It's very interesting. Wow, really steep in this area. Okay, I'd love to zoom in and see what's going on here. Huh. Because I'm seeing a bamboo coral skeleton to the left and then like something really different. Wow. Huh. Wow, so it looks like, yeah, a dead bamboo coral skeleton, which I'm very familiar with, but there aren't any that I know that have that really dense branching. It looks like these might be hydroids or something growing on the dead coral. As close as we can get. This is really interesting and weird. Huh. Yeah, covered with hydroids, really like almost mimicking the branching pattern of a different kind of coral. That is very cool. I've actually never seen anything quite like this. Yeah, I think that's the primnoid that we saw earlier, a similar one. Yeah, these hydroids growing almost as if they're coral branches is really interesting. And of course, lots of other things going on in this rock. Hello. Yeah, the more detail we can get on this, the better. That is very cool. What do we think about sampling this? They should be pretty easy to pull off, I think. Do you? Thank you. Yeah, it's just a really unique, the way that these hydroids are kind of colonized this dead coral skeleton. And look at how intricate the branching is on them. So I'm not at all familiar with hydroid morphology, but the information that we can collect with these zooms, you know, you can see the living little zooids sticking out into the water column. They're almost microscopic, so this is really amazing to see. So I know there are a couple of hydrozoan taxonomists that would be really interested in this. Beautiful. All right, so I'd like to welcome Emily to the geology chair while Jamie steps out for lunch.
Hi everyone, I'm Emily Ash. I'm a marine biologist. Right now I work for NOAA at NOAA headquarters doing communications, but I have a background in coastal zone management and more coastal marine biology.
Okay, so very different from the kinds of places that we've been.
Similar but different, very different. Yeah.
I have the opposite problem. I have almost no experience in coastal marine biology, so when I go for walks on the beach, I'm of no help. But down here in the deep sea, I'm a little more familiar. And I know when I see something a little bit strange that I haven't noticed before, and just the way that this hydroid is branching on this dead coral skeleton is pretty unique. Seems very specific, so I'm wondering if it's not something new.
Well, that's the fun of the deep sea.
Mm-hmm. Mm-hmm. Yes, so I think, like even a snip or a grab should be able to pull some of these off. I don't think they'll be as wiry as the black coral, but I have never encountered this exact thing, so we'll see. And if you're watching this, don't forget you can always tune into stream number 3 to get a few different angles of this collection. It can be really cool to see all of the different dimensions that we have to take into account when sampling something like this. And of course, stream number 2 always has the view from Sirius, so you can kind of look from above down and see how D2 is positioned for this collection. Maybe a little bit lower if you can. Thank you. That looks great. Whew. Excellent collection. Very excited about that one. Thanks, RV. So we've got EX2306-D15-03B in the port outboard bio box, unidentified hydrozoan. ROV watch lead. Do you think we could do a Niskin in this area? No rush. Oh, just caught a crinoid swimming in one of the other cameras. So that one is EX2306-D15-04W, Niskin bottle number 2. very cool diversity of corals and anemones and echinoderms down here. Yeah. Yeah, I'd love a zoom of that and more of that hydrozoan now that I'm looking for it. Oh, a really, yeah, very big one. And so a couple of things that we might want to zoom in on here. This primnoid coral with a really large base. Looks just the white coral with the white base. Just maybe a snap zoom so we can see what it is. I think it's probably Peristinella like the others.
These are so beautiful.
Yeah,
they're some of my favorites.
Oh yeah, I think so. Are kind of the same for me. I spent a long time looking at different Peristinellas in the lab to try to see what species we had. This is farther south in BC. There's definitely some damage, some parts of the branches that are missing polyps. So just having a little bit of a glitch with the camera. Will take a minute to focus. Oh, beautiful, thank you. And yeah, you can see kind of the same really like armored polyps like the primnoid that we saw earlier. And like a really extensive base. I think there's at least 2 or 3 really big branches coming out of this one. Ooh, a worm came out from somewhere.
My favorite.
Wow, yeah, and maybe a view of this really huge Brisingid. If you can get the lasers on it.
Wow.
Yeah, oh wow. Another one just as big, if not bigger. That's wild.
So for those at home, what's the difference between a Brisingid and a brittle star or a crinoid?
Ooh, that's a good question.
Because they all look pretty similar.
I wonder if Chris Ball's around. Yeah, they're certainly, they're different orders. So there's the order Brisingida, order Crinoidea, and so on. And they definitely, I don't know, have a really different body structure, but they all kind of have a similar like central disc, arms radiating out. They seem to have really different kinds of environments and methods that they choose to use for eating or catching prey. Oh, there's a sea star below as well. So yeah, I guess cool. Different modifications on like a theme. I spent a little bit of time with echinoderm samples, but less so. I did see Chris Maud tuning in a little bit. Oh, can we look at that thing?
That's really cool.
We're just about to lose it in the lower right. If we could come back to it. I think it was maybe not. Are we on the move? Oh, there's just I think you're backing up to it now. Maybe the big sea cucumber, I think. Yeah, so back down to the right. It's kind of a dark purplish and just really different from what we've been seeing so far. That thing right there. Is it a sea cucumber? Or is it a nudibranch?
Big nudibranch?
It'd be really big and very different from what we've been seeing. Oh, cool. Oh. I think it is a nudibranch.
It's kind of similar to the really small purple one we saw on one of the first dives.
Oh, wow.
That's beautiful.
Yeah, it like reminds me of the Tritonias, but obviously really different colors. I'm not sure how important color is, or if it's more variable in nudibranchs, but it's really large. I know some of these like to eat corals.
Wow.
Very cool. Yeah, this is definitely a different species or a different morphology from the ones that we've been seeing. Wow. Look at the texture.
Yeah, I really like these feathered fringes almost.
Mm-hmm.
So pretty.
Yeah, these are beautiful. And yeah, very cool. Yeah, I've never seen one like this with the dark purple, almost black. Really distinct dead bamboo coral skeleton to the right as well. But yeah, this very unique nudibranch kind of nested in between these two anemones and the rock. Very cool. Thanks for that. And this gives us an excellent view of this kind of sparsely branched bamboo coral on the way out.
Would that nudibranch be eating that coral?
So I've never seen this one. I'm not sure what this one eats, but definitely similar nudibranchs, those tritonias we've seen feeding on corals. I've got lots of help from the science chat talking about echinoderms. So tube feet in asteroids and brachyngeids come out of an open furrow under the arms. Ophiuroids have tube feet that come out of a hole. Brachyngeids are a kind of sea star. Crinoids and ophiuroids are their own classes. All share a similar body structure in order to advantage current feeding. They accomplish the same goal using different characteristics. And thanks to Chris also chiming in that, also thinking it's a Tritonia nudibranch, really big one. And I would say a different color morph than the ones that we've been seeing. Yes, yeah, we've had some excellent zooms of pretty much everything in this area. So I think when you're good to go, we'll keep on climbing.
You guys didn't collect any rocks while I was gone, did you?
I didn't. Thought about it.
Did you?
And I got distracted by this big nudibranch. No, I don't know exactly what you're looking for. I guess a representative rock from this area. We collected a Niskin and a hydroid encrusting on a dead coral skeleton.
Okay.
Yeah.
Already.
Mm-hmm.
Yeah, if we do see a piece of some of this big blocky stuff, not the conglomerate, a piece would be worth grabbing.
Mm-hmm.
But
okay.
we'll just keep our eyes open. And this might be a tough place to find a loose rock, especially when it's this steep, and we are in a very seismically active area. We're probably within the rupture zone or above the rupture zone of the 1964 Alaskan earthquake, magnitude 9.2, one of the 3 or 4 largest earthquakes ever recorded. So things do get a pretty good shaking here now and then.
Here we're getting a good view of one of these clear sea cucumbers we've been seeing a few times.
Watch lead. Nice little sponge.
Indeed.
Oh, wow.
Wow.
That looks enormous.
Really does. Yeah, it looks like that central area would be basically the lasers. 10 centimeters. That's really big. Yeah. Very impressive.
Moving past a little what looks like a shear zone here, you can see the vertical joints and a little low spot or crack where the sediments filled in, marking it, but another some evidence of the kind of deformation that these rocks have undergone. I wasn't sure if we were going to be able to see that kind of deformation. I didn't know how, before we got here, how broken up these rocks might be. We could have been ascending a fairly intact face, for all I knew. But we see at least local and deformation that's breaking up the rocks here. So, again, another indication that these have had a fair amount of baking and faulting in their history.
A nice view of a sea star and a bright red shrimp.
He's up at attention.
Mm-hmm. Having his moment of fame.
Yeah, let's head on up, see what we can see.
This is a beautiful view of this coral. You can see some of the tentacles are fully outstretched, and others are retracted, probably as it feeds on things passing by in the water column. It uses its tentacles as little arms to grab things, and it brings those things that it catches into the center where its mouth is, its feeding structure. It's a great look at all this.
And I think I'm seeing little dark bands on the stalks. With spines between
This
the
one
tentacles?
has, yeah, spines between the tentacles. A little bit more pronounced than the other ones we've seen. Another one of those really large bracyngids. And some more flytrap anemones.
Wow, those are enormous.
Yeah, they really are. A lone sea star in the sediment. A sea star or a brittle star? Let's see. Looks like a brittle star. Yep. Some beautiful corals.
And that is a...
Anemone.
Anemone. Oh, he's
Yes.
got very ornate tentacle structure.
Yeah. It's very peaceful to watch them move around in the water column, in the light current. Yeah. It's always amazing to me how much detail these cameras can capture. It's so beautiful. Really special to be able to go into these very inaccessible environments and get such a good look at things.
At ROV Watch Lead, I'm looking at a rock over in the bottom right corner of this view that might be a good one to grab. Sort of a roundish boulder coming up towards the lasers, not overly big, but I think it looks loose. Just blown to the right, now on the right laser beam. Yeah. Never sure how big things are until the lasers get on him. He's a little smaller than I'd like, but if he's grabbable, it'd be worth getting him. Yep, he's connected. Yeah, alrighty. Well, let's keep going.
If we could go back to that anemone we were just looking at, it looks like there's an interesting shrimp that's been crawling around right under it. I'd love to get a zoom on that. Thank you. Just below the anemone, right to the left, on the left side of the screen right now. With the bright red spot on its head, yeah. Its eye caught my attention. Interesting. Very sedimented, very dirty.
Yeah, we saw a crab at the start of the dive who was very muddy looking.
Interesting.
I don't know if that comes from just crawling out from underneath the mud or if they like to keep it as kind of a hiding strategy.
I love the way the light catches his eyes. There's a suggestion in the chat, a cragnoid shrimp. Thanks for that zoom.
Onward and upwards. So I mentioned these rocks might be part of what's known as the Yakutat Terrane, which is my best guess for what these are at this point. The Yakutat Terrane being a fragment of the crust that was originally sitting off the coast of current-day Washington and Oregon, and was essentially broken off and carried northward by the Pacific Plate until it has encountered the Aleutian Subduction Zone, where it's now in geologically slow motion, but nonetheless crashing into North America and the North American plate. And it maybe sound a little fantastical. How does a big chunk of crust this size, it's on the order of 500 by 100 kilometers in size, and has partly already subducted or been accreted onto the southern edge of Alaska. But how does something like that get torn away from the coast? How does that happen? A modern day example is perhaps not exactly the same process, but might have been, is Baja California, and the way it is broken away from the Mexican mainland after North American plate overrode the old spreading ridge. That spreading ridge is now in the middle of the Gulf of California and is in the process of tearing Baja California and most of Western California away from the continent. Those parts of the continent are now part of the Pacific Plate and will eventually make their way north, or may eventually make their way north. But that is a process by which most of Alaska, large parts of the southeastern panhandle were assembled in little bits and pieces of crust that were carried here by the Pacific Plate.
So just coming back from lunch, what did I miss? See a couple of really nice big primnoid corals.
Couple great big brasingids.
Okay. And have you been looking for a rock to collect, keeping an eye out?
Yeah, we tried a rock, but it was stuck.
Oh, okay.
So one of the reasons to grab a rock is to verify that story I just told. The sandstones in particular can carry a mineral called zircon, which are very resistant, and they can not only survive a long sedimentary transport, but they can survive a good deal of metamorphism, and geologists can separate those out, get a handful of those zircon grains, and actually date the individual grain. And they, in so doing, you can get an idea of where they're from.
Huh. This might be one of the eel pouts that we have, but it looks a little bit different to the ones that I'm used to seeing. Definitely a different kind of lighter color, and now I'm not so sure. So it's always important to capture these nice tight zooms. You can see all sorts of diagnostic features on these animals. You know, every ray in the fin of that little fish if you wanted to. And that can give us important information about what species it might be. So even if we can't figure it out right now here live, we'll be able to look over the video later, compare it to other specimens that we have, and get a species identification on this one. Look at that line of dots going just along the side of the body and then up along the dorsal fin. You know, you can see features with this zoom that are just amazing. Okay, and more of these lily pad, these black corals, quite a few really big ones. There's a group of 4 really big flytrap anemones to the left.
Taking over that rock face.
They really are, and there's more coming up. Another really unique looking anemone that's kind of folded in on itself a little bit, probably in the genus Actinernus. This white coral is a primnoid Peristinella that we've been seeing in a couple places. This looks like a Actinernus that's just a little bit droopy. Seeing small white encrusting sponges every now and then on the rock, some larger tubes. It could be serpulid worm tubes. Just all kinds of life on these rocky outcrops that we're seeing emerge from the mud. ROV, could we pan over to the right and look at that long sea pen sticking out of the mud? Not sure if we got a great look at this one earlier. This is my first time seeing it on this dive. Probably a sea pen in the genus Baltacinus. So sea pens are related to the corals that we've been looking at, but instead of making a large branching skeleton, they make one really long axis. Often they have modified kind of swollen ends at the bottom called the peduncle that allow them to anchor in the soft sediment, and then they have these sort of almost, leaf-like appendages sticking out with a few polyps on them. You can see, looks like most of them are retracted, but if we go a little bit further down, I think I see one that's open. There it is. Just one. So it doesn't have all of its tentacles out, or if it does, they're a little bit damaged, but you can see 8, which indicates that they're grouped with the octocorals. 8 tentacled polyps on these corals. You can see little spikes kind of emerging at the base of the polyps that also indicates to me that it's in the genus Baltacina. That's great, thanks for that. This is really excellent, actually, this close-up all the way down this really long coral. Brittle star wrapped tightly around close to the bottom, and then you see the polyps disappearing to this modified portion at the end. That kind of swollen at the bottom is the peduncle buried in the sediment. Very cool, thanks for that. A small crab.
A muddy crab.
Yeah. Looks like it's less than 10 centimeters. Maybe one of the tanner crabs, a small one. Like a juvenile of the larger tanner crabs we've been seeing. Ooh.
Well,
A little too close. And one of those carnivorous sponges just to the left. But some really large shrimp. I think I'm seeing a stocked barnacle. We've got good close-ups of all of these things, so we'll just kind of scan as we move up this feature. And lots of black corals and Peristanella on that rock to the left. There's a dead bamboo coral, quite a large one, the skeleton of it just going out of view. Wow, those are pretty big. Oh, could we zoom in on this sea pen? It looks a little different from the one we just saw. Thank you. So I'm seeing just a different pattern of branches kind of coming off of the side of this one. Ooh. So yeah, instead of being on kind of a longer row that's almost leaf-like, you see individual polyps coming off of the main axis. Definitely different from the one we were looking at. Just checking to see if anyone in my records is interested in a collection of this one. I think we'll just collect this really good imagery. I think we should be able to tell what it is from this zoom. It's also just beautiful with the kind of faded out marine snow floating in the background. Looks like there might be a couple of them coming up. And this is this brittle star that kind of looks like a basket star, maybe is kind of something in between. I think I heard Chris Ma refer to these as snake stars, so they're like basket stars, but they don't have the highly branched arms. They're just really long, kind of curling branches. Wow. Very cool. I'm not seeing anything that's standing out to me as a sclerite that you can see, so possibly they have really small ones, or some of these sea pens have none at all. And that just got just close enough to those polyps that got them to retract there. Oh, and I maybe am seeing a little thin kind of layer of very small sclerites lining the outside of each of these tentacles. So features like that will help us to identify it. I think we won't take a sample, just a really detailed video here. So I have an ID of Euryalidae for the ophiuroid on this sea pen. Ooh, very snake-like arms. So the central disc of this one really looks like a basket star to me, with those, you know, really, those like thick lines, kind of ridges going along towards the center. Aha, we have an ID of Asteronix leveni from Chris Ma. Thanks for that, Chris. I think those are two more of those long, thin sea pens in the background with their own associated Asteronix, the kind of brittle star, the snake star on them. Really nice branched bamboo coral on our left, and more of this one of the black corals that we've been seeing. Lots of this Actinonus anemone. Sorry, Actinernus. Maybe something like Actinernus nobilis. Just a snap zoom of those sea pens as we go by would be great for our records. They're hard to tell apart from far away. That's great. Okay, thanks for that. Definitely more aligned with the second sea pen that we looked at. So I think we've had really nice imagery of all of these. We can keep on just kind of cruising on by and documenting them.
Does that look like a loose rock over on the right? Maybe lying there?
Over here?
Yeah.
Looks like we're just pausing to do a pilot switch, giving us time to really take a good look at this rock outcrop covered in anemones, black corals, and a mushroom coral, as well as some really small encrusting sponges. Nope, I don't think so. We are looking for a rock, though. Jamie thought he saw one, maybe that looks a little bit loose.
I wonder if it's that, well, it's hard to say. It's on the far right. Well, that one does look loose.
And that definitely came from up here, I would say.
Yeah.
So just to the right and below the lasers, very close now, just below them. That.
What do you think of that slabbier one over to the right? Just up and to the right? Is it going to be too big? And is it loose? It's key.
Looks like there's
sometimes they fool me, but it has a look
something
of
under it that might be loose.
Yeah. Well, that's true. Yeah.
Actually, if we could zoom in just a bit to the right and above the lasers. Yeah, kind of scanning, seeing if there's something under it that might be a little smaller. Ooh.
Yeah.
Maybe?
Yeah. He might be too long in the end to get into the box, but perhaps that one that it's resting on, yeah, is definitely sitting there loose. If we could zoom in. I think that would be a good one if it's not a mudstone, but I see parts of it have pretty good varnish on it.
Yeah.
We're looking at a broken surface, which is something we don't get very often down here.
Can we set up to do a test, see if it'll hold?
Okay. Sounds good. Yep. And there it goes. You've started a turbidite. Yeah. Yeah, go ahead and see if see what that's like. It seemed like it was the same thing, but it might help you break off that other part too. Sure. Yeah, okay. Yeah, we're out of that, I think we're out of that real hard rock, so that's okay. Yeah, no problem. Yeah, well, it turns out we're probably not on the rocks I want to collect anyway. These are younger and sitting on top. Thanks, though. It was good to see that. Sometimes it's good to just stop and hit a rock, see what it is. Really nice view on Cam 2 of D2 from Sirius coming up this wall. We're getting to the top of this rocky face. Crumbled.
So a couple of smaller corals. Looks like could be little bamboo corals.
Yeah, it's hard to see. These look like the more what I think are lithified rocks, but I don't see, once again, anything loose unless that one kind of lower right. Right now below the lasers might be loose. Might not be, yeah. Oh, yeah. It'll be also interesting to see if these are crumbly also, or if...
I mean, I'd suggest that one, but there's giant anemones on it. Don't know what we'll do with them.
Yeah, well, there's also that piece above this bigger one looks loose as well. It is loose. Got to be loose. Yeah, let's give it a poke test and see if it's loose and hard. Yeah, right below the lasers. Not moving, maybe, but hard. Yeah, how about the one just above it, the kind of wedge one right behind the arm right now? On cam 1 anyway. Leaning up against, yeah. All right.
See that one.
Well, yeah, the one just to the left that's sticking up, left of the one you tried. It's a little ledgier, but I feel like it's connected, but you can give it a knock. Yeah.
Mm-hmm. Wow.
Mm-hmm.
Okay.
Yeah, it's good to know these are hard rocks, though. Does that wedgie one look connected, did you say, from another view? Or are we trying to move around and see if we can get it? Also, there might be one up in the extreme right. Yeah. Mm-hmm.
Yeah. That one there.
Yeah. Oh, yeah, that one is connected. The other one is connected, I can see now. And this, sure, yeah, give it a hit. Does it? Yeah. Yeah. All right. Well, let's keep going. Yeah, we have big competition here. The subduction zone. That's probably doing a pretty good job of rattling anything that's loose on down the slope.
Oh, yes, always. Thank you. I was looking up for rocks. I almost missed it.
Usually Chris Ma is the first one to speak up.
Mm-hmm. Okay, not familiar with this one. So we'll collect this nice, the best close-up imagery we can and see if Chris pipes in, but not familiar with this one myself. Beautiful. That's great. Not getting any chatter in the science chat about possible IDs, so we'll just stick with the zoom that we have and continue on up. Thank you. Some kind of a yellowish worm poking out from under that rock. Can we zoom to the right, that white thing, to the far right? Yeah, still to the right a little bit. What is that? Oh, interesting. A different kind of sponge than the ones that we've been seeing. Cool. Some kind of, I don't know, actually. Assumed it was a glass sponge from far away, and now I'm less sure the closer we get. Yeah, this is great right here. Just kind of collect what we can and...
Now, how do you tell if it's a glass sponge by looking?
Well, I'm looking for any indication of that 6-rayed geometry, so any of those regular patterns. This is looking a bit more like a demo sponge to me, but this one is actually pretty hard to tell. It's not as thin as the other ones, and there are glass sponges that get like this. Yeah, so we'll have to do kind of a detailed review of the video later and see if we can pick out any characteristics that can lead us one way or another. I'm leaning more towards a demo sponge for this one. That's great, thanks for that. And of course it always helps to see more, see more individuals, maybe farther along in their growth. You can see different characteristics than you can on juveniles, so and maybe a different growth form. Yes, that's great, thank you. You can see a couple of attachment points, I think. Very cool. I mean, it's a pretty large one. It's really interesting. The ID on that sea star we saw earlier is low faster. Thanks for that, Chris. There's another. Actually, can we zoom in on this this sponge now too? I think there's two different kinds. So it looks like we're seeing these like really large folds is perhaps different from the kind of branching one that we see coming out at the bottom and the top. So it could be two different kinds of Faria sponges in this view. And this is a glass sponge that I'm really familiar with. And you can see when you zoom in a really clear pattern. You know, they're almost like a square network where those 6-rayed spicules intersect with each other and make a really regular pattern. But then, yeah, we have this branching structure coming out from the bottom that has the same general pattern, but obviously really different in the way that it all comes together. So I think there's two separate Faria sponge species right there. Both have these shrimp associated with them. You can see some bigger ones, and the more you look, I think I start to see smaller and smaller ones. So one of the reasons we're interested in these sponges is because of the habitat that they create for other animals, and you can see a nice example of that right now. That's great. No, I think we're good, thanks. Yeah. Oh, thanks for that ID, Chris Ma. Low faster for Silager. So sometimes, yeah, we get enough detail from the close-ups that we can even take it down to species. That's pretty impressive. These rocks are just covered with a lot of anemones. A couple different kinds, those big flytrap anemones and the purple actinernus. Lots of liliopathes, the black coral. I think I'm seeing a really big branched bamboo coral in the background, and some of these sea pens poking out of the soft sediment in between. While we wait for a ship to move, another Asteronix leveni, the snake star, tangled up as it climbs this sea pen, Halipterus or Balticina genus of sea pen, just to get a little bit of an extra leg up into the water column. Apparently the central discs on these really deflate when they're collected, so they're not as beautiful as they are in situ, and they're full of water kind of doing their thing. Just a really different central disk than the brittle stars that we've been looking at. Another one of those very cool carnivorous sponges below the smaller bamboo coral there. And up on the other rock in the background to the right, this really nice branching bamboo coral in the middle. Been seeing a lot of dead stalks of similar ones, so it's nice to see live, healthy looking colonies. A really big, bushy black coral. Looks similar to the one that we sampled earlier. Yeah, if we could get a snap on that sea star as we're going by. But in general, we'll keep on climbing. certainly looks similar to me to that low faster we saw earlier.
Don't those have to be loose rocks that are sitting around him?
Do you want to give him a nudge?
Yeah.
Yeah. If one of them has to be loose, I would think. Maybe the far one?
Maybe. Yeah. These, well, perhaps the one to the upper right? Yeah. I don't know. They look like they've got to have a random orientation, but maybe they don't. But one way to find out.
Mm-hmm.
Oh, is that coral's going to be in the way, I think. Oh, we're going to be able to reach over him here. Nice. All right.
Ooh. You want to check the one beside it that the sea star's on?
Yeah, I wonder about the one the sea star's on, but
Could we nudge the rock that the sea star is draped over? Yeah. Thanks.
there we go.
There we go. Yeah, we don't need a collection of low faster today. Perfect.
There he goes. Nice. This was something he was not expecting to happen today.
Interesting that low faster is in theory the same species as the shallow ones you can see scuba diving. Low faster fossiliger. Ooh.
Softer than we thought. Okay. Interesting. Well, let's hold on to it. Yeah. Now that we've got it, let's... Yeah, no, that's all right. Let's just get this one. And it'll actually, for purposes of looking at a younger rock, would be just fine. No, that's fine. Yeah, let's just grab it. Oh well. I've seen worse. ID EX2306-D15-05G, starboard forward rock box. Yeah, that's the thing. The big blocky lithified ones are going to be too hard to pick up if they're still sitting here.
Oh, this is excellent. We don't often get this view of the sea stars that we see. So you can see the little tube feed poking out in those furrows along each arm. Lots of armament, different spikes all along the outside. Very cool, and that sea star will use their little tube feet to attach to the rocks and flip itself back over. Ooh, I don't know, or might be a little crustacean on it. Yeah, might not be able to tell from this angle. Yeah, you'd never even notice that, I don't think, on a regular camera.
I'm reaching out to find the rock.
Yep, yep. It's already got its tube feet attached to one point. All right, so we'll continue up. See a really big stalked barnacle. We had a nice zoom on one earlier, so we'll keep on moving up. Lots of anemones. It's another large brisingid. We've had quite a few brisingids zooms if we're good to move on. Just kind of document what we're seeing along the way. We can, do counts as we go up and get estimates of the overall abundances in this area.
Robert Carney with a geologic note on the echinoderms. Their hard parts are optically similar to calcite crystals.
Oh, interesting. I do know some of them have little calcium carbonate ossicles in them. I think more sea cucumbers. Some really nice big lily pathies, these black corals. More of the sea anemone, the Actinernus. So this looks like hard rock, but would you say it's probably more of that
I think so,
mudstone?
yeah. The mudstones, yeah.
Wow.
But you can see it has a kind of a much lighter appearance and by and large, and is not so fractured. And we've also not seen any of the conglomerates for a while, so we've maybe even moved up above that interval.
Maybe just a quick snap zoom. It's kind of hard to tell. Thank you. Yeah, really small farreid sponge. So you can see it's just starting to fold in a little bit, but it's got that really thin skeleton. That's great, thanks. So we've got a bit of ground to cover. I think we'll just kind of fly over most of these and, record abundances and see if we can make it a little bit farther up the slope of this feature. Oh yeah, I think there might be a couple. Yeah, a snap zoom on the squat lobster as we fly over would be great. But we don't need to stop.
Oh yeah, there are a couple hanging out there.
Kind of whitish looking.
Sticking out of their caves.
Kind of an unusual angle to catch these. You can see, yeah, the lighter white color. A lot of the squat lobsters we see are kind of a pretty deep orange. It's got lots of, looks like maybe associated fauna growing on the legs and the claws. I think that's great. Well, I can't tell if they're from the crab itself or if they're little encrusting fauna like hydroids maybe on top. They don't look like they're branched. Yeah, I couldn't tell you. Ooh, a different kind of black coral. We saw these in the beginning. Those two larger, kind of brighter orange ones are bathyes. Nope, just kind of calling them out. I'd like to cover a bit of ground if we can.
So we've come up about 100 meters up the slope from where we started.
Seeing lots and lots of corals. Just very nice to see, especially when we first landed, and even right now there's quite a bit of sedimented areas, but everywhere where the rock is clear enough, it seems to have something on it, an anemone or a sponge. Yeah, nice mushroom coral. We had a good zoom in on one of those earlier, so I think we'll just document it as we fly over it. That's right, we're getting, every now and then pretty moved around up here in the control room, and we have Sirius in between the ship and D2 kind of taking the brunt of those movements and keeping things nice and steady below. We've been getting some amazing imagery from D2 today. Oh, interesting. There's a dead coral skeleton. It looks like the skeleton from a primnoid, probably that Peristinella that we've been seeing around. Little small sea star, or brittle stars. But yeah, it looks like a piece of a rock, maybe some of that mudstone, eventually
Was
kind
just
of crumbled and moved.
Crumbled on him.
Yeah. Huh. Sure, thanks. I can't tell what that is. Really long. Huh. So that looks like a really small, maybe a bamboo coral of some kind, and maybe almost looks like a piece of a kelp or something. It's so smooth. Huh. Yeah, I don't know. I think we're okay. Could be a really long, I don't know. It's not fishing gear. It's not a coral, so I'm not totally sure.
Yeah, this is not just big exposures of this soft mudstone. You can tell it's a lot softer. It's easily bored, and so we see lots of borings.
Yeah, I think we've got some nice imagery of those snake stars on top of these sea pens. Looks like every one of them has an associated, once they get to a certain size, almost everyone has a snake star on them. Asteronix leveni. Lots of these very cool cladorized sponges. And these white and purple anemones just everywhere. Kind of right from very close to the beginning of this dive, we've been seeing them ever since. Lots of the flytrap anemones in the background as well. Will do, sounds good. Yeah, I think we'll just try to cover some ground, and we've got nice zooms of most everything we've seen here. A little bit of a depression. Oh, a little crab coming out of that hole. It's always helpful. Oh, really small. Well, I would just be guessing if I assigned an ID to this one. But very cool to see them coming out of those holes. Now we at least have a little idea of what might be in there. Oh, that's nice. We haven't seen one yet on this dive. A hyocrinoid. So this little yellow stalked one is coming into view. Related to some of the crinoids that we've seen in other dives that don't have a stalk, that kind of stay more closely associated with the rocks. This one has a bright yellow stalk and arms. Can be very cool. So you can see at the end of that long stalk, 5 separate arms, and the mouth in the middle. Each of the arms has these little pinnules at the end, kind of increase the surface area that the crinoid uses to grab things out of the water column. Wow. With this level of detail in the zoom, I think we can, almost count individual segments on the arms. These are things that can help us assign a species level ID to this. Yeah, they're very interesting. I wonder who made those.
All the footprints.
Yeah. Sure. Yeah. Let's just take a little bit of a closer look. Huh.
That's really interesting.
That is interesting. Not sure what to make of that, but always important to document things like that we don't know. Beautiful. Yeah, just everywhere now. Now that you look.
Robert Carney says spider marks.
Oh, interesting. Possibly a sea spider? I don't think I've ever seen one move. I think that they can. I think they can detach somewhat, but I've never seen one. I think they hold on a little bit with some of the tube feed, and there are also, I think, modified plates underneath the central disc that help them kind of adhere to the rock. Wow. Beautiful bamboo corals. With one in the middle that's missing most of its tissue. Ooh. Flytrap anemones reacting to our presence by retracting and balling up.
And I'm suspecting we're seeing a bigger population and a denser population with the ones that are anchored to the harder bedrock. You can see it.
Sure, I guess we've seen like smaller individuals, as soon as they get to a certain size, the mud rock just can't support them anymore.
Right.
Or it shifts because of
or
the...
it falls down the hill in an earthquake. Here you see little pockets of that mud, but I believe those are the darker colored and smoother rocks that aren't bored are the harder but unsampleable bedrock.
Unfortunately, it looks like you could sample it, but they're all the ones that we've nudged are very firmly attached. A really beautiful bamboo coral here with a wide range of these very cool cladorizid sponges in the background.
That one's pretty tall.
Yeah, there's a lot going on here. Brisingid. Yeah, we've seen some really huge ones. Getting into more black corals over here, I can see some skeletons that are kind of mostly dead and without tissue, just like little tufts of kind of the orangey tissue still attached. Lots of these Actinernus anemones. Those little white-stalked things are carnivorous sponges in the genus Cladorhiza, probably Cladorhiza corona. Oh, we've got a good zoom of this one. Oh, okay. Yeah. There's some. Appreciated. It's another one of those stalked crinoids, the yellow hyocrinoid. And a balled up flytrap anemone. Lots of these carnivorous sponges. Every now and then I see the legs of a brittle star kind of hanging out on the rocks or just under them. Wow, quite a few really large bamboo corals here. Makes me think the records that we have from farther up at shallower depths for Gorgonian corals are probably either bamboo corals or that one to the left is a really big primnoid, Peristanella. Actually, it would be worth a zoom on that big white one just to the left.
Ooh, wow. In the distance or the...
I think that there's a little Panikia sea cucumber to the left as well.
Mm-hmm.
Ooh, Yeah, so you can see the darker skeleton underneath, kind of covered in really dense sclerites in a lot of places. Some of it seems to be missing some tissue, but there's a layer of these hard calcium carbonate sclerites kind of armoring each individual polyp and the branches. So this is a Peristinella. I've seen a couple of these, but this one is, for one, really large. We're getting a great view of the polyp morphology. Might even be able to zoom in on this at afterwards and kind of identify it, maybe even to species. Wow. So the general morphology of the polyps, but also the placement of each of the sclerites is how you tell the species apart in this group. You can see brittle star arms entwined in the branches. Yeah, it definitely looks like this one has seen some kind of damage. It's beautiful. Thanks for that. Really nice. Interesting, yeah, that it's not branched all in one plane like you often see them kind of in a fan shape. This one is going in many different directions. Whether it's been damaged or exposed to really different currents over its lifetime. Oh, there's a sponge we haven't seen yet. Yes.
Oh, interesting.
So a lot of these are glass sponges in the family Rosselidae. Few different species that it could be, but and it also has a really different, it has that kind of side projection coming out. Oh, beautiful. Look at that. You can see some associated fauna right there, and lots more the closer you look. All those little tiny dots are possibly other smaller things. And yeah, you see this like tufts of long spicules sticking out of this sponge. So it's a bit of a different shape from what I'm used to, but probably one of the boot sponges in the family Rosselidae. We haven't seen one that big yet, but a snap zoom would be nice. It's a Faria. Yep, definitely a Faria sponge. So another glass sponge in that genus. There's a few different species it could be. Oh, and there's another one maybe up, another white sponge, just up and kind of to the right. Is it? I couldn't tell. It looks like it's on a stalk, which would make it very interesting. Oh, cool. So we were... no, it's okay. Yeah, we can keep on going. I think, yeah, we did sample a stalked sponge on an earlier dive. Has the same general morphology. So we could potentially have an ID for that one. Wow. Yeah, some really big corals. Look at that big one. Sounds good. It's huge. If we could get the lasers on it to get a, yeah. There's somewhere. Wow. Huh. This is one of the bamboo corals. There's maybe a juvenile of the same kind just below. This one is a huge, really healthy looking colony. Placed like a little precariously on that rock. Mm-hmm. Hoping it's not mudstone and it's something a little bit harder that can kind of stay put. And of course more of that Faria sponge on the bottom, and lots of these actinianus anemones. Beautiful. Look at the snake star just kind of frozen there.
So would that be on the order of 30 to 50 years old, or is that something this size even older?
Bamboo coral growth rates, I actually don't have a good frame of reference for, so I'd just be guessing. Every species is different. They haven't all been studied. I would imagine bamboo corals have, because they leave behind such those really big stalks and bases, which is the part that you would need to do an aging study.
Right.
But I've never read anything about bamboo coral ages. And of course, even harder, this one, it's not totally clear what species it is. So even if I knew a general bamboo coral ages, it's hard to tell based on the fact that we are not totally sure what species or maybe even genus this one is. Beautiful. Well, it's not. If it was a well-described species, then yes. But because the taxonomy isn't quite there, I can't. But if it were possible to do it from a zoom, this would do it. You can see pretty much every detail of the polyps that you would need. I'm not seeing a lot for sclerites. Every now and then I can see kind of long lines farther down the neck of the polyp. But you can see really well in these ones. They're way less armored, certainly, than the primnoid corals that we've had a zoom on. You can count very easily. Each of them have 8 tentacles. That's how we know that it's placed within the Octocoralia. Each of the tentacles has little pinnules coming off of them to kind of increase the surface area, which we see, in the echinoderms that we looked at, but also many coral species. It's just beautiful. Wow, I love that shot.
Just incredible photography.
Beautiful. That's interesting. Quite a bit of damage to that colony. Can we zoom in on this other sponge in the middle? Yeah, the white one right by the lasers, right there. Okay, interesting. Thanks for that. No, that's good. Yeah. Ooh, actually, to the left, there's like a branching coral sponge right there. Yep. Ooh, beautiful. This is a different morphology of the Faria genus, maybe, although now that I get a closer look, it's really hard to tell, but similar sponges I've seen to this morphology are in the same genus. But yeah, they have this kind of branching morphology instead of making these big kind of lobed, but overall like globular shape. They make these long branches. It's something a little bit different than what we've been seeing. That's great. Thanks for that. Wow. No, that's okay. I think we'll just kind of document them from afar. It's interesting to note that that one has a bit of damage at the base, kind of emanating out. Some more of that Paristinella coral just to the left, and Faria sponge. Oh, I think an anemone probably, just the back of one. Yeah. Oh yeah, I think I can see little tentacles coming out actually. Yeah, that's fine. Wow, just I've never seen. I don't think I've ever seen this many of those Actinernus anemones. They're just everywhere. Sounds good. Okay, we're definitely getting into more of an abundant area of these branched bamboo corals. Rocks are surprisingly hard to come by around here. Every one that we've tried is either firmly attached to the substrate or it's mud rock.
Yeah, these look like the mudstones.
Oh wow, yeah, quite steep in this area.
I think those are going to be soft mudstones. We have a bet. Yeah, we're looking for something you're going to bounce right off of.
Definitely like a deflated anemone that we just passed by. So the one up to the right is completely retracted or kind of balled up. Lots and lots of this purple Actinernus. I think I see a barnacle towards the bottom, maybe a stalked barnacle. We've had excellent zooms of all of these things throughout the dive. And look at the density of these bamboo corals here. Those are just huge. And then in the soft sediment, we have these really interesting sea pens that are really often associated with Asteronix leveni, the snake star. They're really long, kind of snake-like arms. There's a dead skeleton of a Faria sponge right at the bottom. Maybe a snap zoom just to kind of look. Thank you. So these have really delicate skeletons. There's still lots growing on them, but they don't seem to last as long because they're just the one layer thick. Some of the reef-forming sponges have really strongly fused kind of scaffolding for skeletons. These ones are very easy to break. Wow, that one's giant.
Yeah, yeah.
Lots of stuff. Oh, cool. Yeah, stalked sponge. I've seen similar ones to this in areas farther south. We call them, I think we called them sea orchids or sea lilies or something like that. Also interesting, something encrusting on this rock. Yeah, I'd like a zoom of both of these again. So we have these sponges in the ID guide on board as Caulophagus. No species name on it, and it's possible that it's undescribed. Oh, you can see really tiny crustaceans and maybe even a little shell associated with the top of this sponge. Sounds good, thanks. Just not really sure what I was seeing. Ooh.
Oh, what is that?
Ooh, I don't know yet. Oh, wow. Looks like really small. They remind me of little corallomorph anemones because they have those balls at the end, but I've never seen this kind of density. Really cool, extremely dense, very small ones, kind of almost overlapping each other in parts. So they're not zoanthids that I know of. They look like little tiny individual anemones. For whatever reason, just loving this one rock.
They seem to like to live together.
Yeah. Beautiful. That is very cool. Definitely keep an eye out for more of those. But yeah, they seem to be super localized. I'll definitely keep an eye out. We have had a request for a collection of those small anemones. I think that rock was too big and had too much going on. I don't know. Wondering how well that would work. I think a rock that it's growing on would be ideal, so I'd like to keep an eye out as we go forward. Yeah. Ooh. Yeah, that's the things. Yeah, and I don't know how well they would do to suction off. They seem pretty... Yeah. Because if this is mudstone, then it'll kind of crumble, but we might get enough. Some very cool glass sponges along the way as well. Yeah, a few different kinds of sponges in this view. I think we've seen... Really? Ooh, is it not too big? Okay. Would you want a piece of that?
Sure.
Yeah. Do you think it would fit in the bio box, or are we looking at the rock box? Awesome. Yeah, let's do it. You've got starboard open. Mudstone.
Oh dear. Yeah, there it goes.
Yeah. Yeah. Okay. I wonder, do you think there's a piece that you could lightly pick up that might have one or two of those anemones still on it? It's Oh, I see a little bit. Wonder about what about you see that little chunk?
If you grab that
right.
Corner
branch thing,
but I don't see
if
anything
you
on
tilt
it.
the camera down again.
Where are you going, pilot?
I'd zoom out partially.
It's right there, but I don't know if there's
triangular
anything
one?
to grab.
The
See that
science
branch
chat also
thing?
didn't expect that, so
I hit it with the
appreciate
toe of the RV and
appreciate
it's
the
pretty
try.
stiff. I think now the chunks are so small that they're going to be like too difficult for me to grab.
Okay.
Like I'm just going to keep mangling it. I'm
You
concerned.
can see what's in the wrap box. That's what we ended up with.
You know, I think we leave it.
Okay.
I think we're better off spending our time.
Yep.
You don't think you can cradle it?
No, because it's too small to fall between the jaws at this point. All right,
What
come out.
about what
Steering
about a
drops.
section?
Maybe. Yeah. Let's see.
Do you want to do
See if I can come up with
steering
it.
hydraulics for now?
I don't know.
And you get extra points if you can get one of those little red shrimp in it. Also on our list. But I don't see it now.
It's too dusty at this point. I think we should keep moving.
Yeah. Let's
Actually,
see some more on a
we might see a
quickly.
different. Yeah, there might be another opportunity.
All right, I think they're all going to be
Steal
on the
my
same
camera back,
kind
pilot?
of stone.
Okay.
Yeah.
Maybe if we see him on the same kind of stuff, instead of using the arm, I'll go right with for the suction, and then
Okay.
maybe I can use the end of the suction nozzle to kind of like break the mud apart, and then maybe that'll get him in.
Looking down 70 degrees, pilot. It's a good thing we put in that easy step. That's perfect.
I'll keep moving here. Keep an eye out for you. I got the mudstone lesson.
Sorry?
Yep, still mudstone. You can tell by the way it gets bored, even in places where it has a little bit of an iron stain.
There's
Ooh,
another one of
another
those.
one of those big sponges.
Coming up.
Ah. There's only one way to be sure it's mudstone, and that's to watch it crumble before your eyes.
Yeah.
Yeah.
I bumped it at the wing of the ROV, so I thought it'd be more stiff. It got me good.
Huh.
I'm scanning for your little guys here.
That's all right. We don't have to look too hard. I'd rather cover a bit of ground, I think.
We can, whenever you're ready, pilot. Yeah.
Understood.
Keep
These,
pushing
it's
up.
just a really great opportunity to
Coming up with
document the abundance of these corals. We've got excellent zooms on lots of them, so we can be pretty confident about
And
the
if you're going
ID.
to keep pushing,
and now
we can go ahead and get
just
another
kind of
20 meters.
documenting the numbers that we're
Send
seeing.
it.
Let's do standby 1,
It's
035.
incredibly useful.
Please, Nav, thanks.
Bridge over now.
So, like, yeah, several
Hey,
different
Bridge,
kinds
I'd
of
like to put
black
another
corals
move
as
in,
it is
please.
to genera
20
there.
meters,
Another
bearing
really big
035
bamboo coral
degrees,
right in front of
please,
us.
To the
0
right of
knots.
this that just landed, is that the one they're after?
I don't see it.
Just went off screen right.
Oh,
Good
sorry, I
copy.
missed
Thank you,
it.
Bridge.
They're often associated with corals,
Send it up on 035.
and we've had a
pilot.
request from a researcher who's looking for the
20
genus
meters in this direction.
Lebius.
There's one there as
Oh
well.
yeah, a really
Lower
small
center.
one down there. Might be hard to get from this angle.
Which one do you want to closer
Looking
look
at that little shrimp that you can see right through the
snap in.
bamboo
Go
coral.
ahead. In.
Let me know if you want more time here, pilot. We're going to ship to a stop.
No,
That does look like the target, so shrimp like that one. I don't know if we can get at this one.
Oh, you want to collect a shrimp?
I do.
Oh, okay.
You almost got it.
Oh yeah, it has a really
Yeah.
short rostrum.
All right.
I'll
Different
call it. I'll
from
stop.
Yeah, sounds good. Thanks.
some of the maybe
Bridge over
pandal
now.
shrimp that we.
Hey, Bridge, we'd like to
I've
easy
seen.
stop that move, please.
Watch it. Do you want me to try to collect this shrimp?
Thank you, Bridge.
Yeah, I think that'd be great. There's actually one at the base of the other lighter colored bamboo coral too. Might be easier to get. See
Right,
it?
right here.
Yeah.
Gotta be
Snap
quick.
it. To confirm.
Yeah, snap it. I'm assuming you want a suction sample, these two?
Yes.
Okay, come out.
Yeah.
Oh.
Actually, there's two of them. Slightly different color morphs.
Ready for camera?
Yeah.
Hydraulics,
So I
right?
spent a bit of time before this dive
Jar
looking
light,
at
you're on
the
jar
differences.
6.
And
Copy.
I think
Jar
I can
light
spot them.
Okay.
Yeah, I'm pretty sure these are the targets.
Section 6. We're going to go to 1.
I'm ready for you to draw out.
We're going to have to sneak up on them.
Yeah, I mean,
Up a
obviously
little
they
bit.
can
I'm
swim
just going
pretty
to draw
fast,
out.
so we'll probably have to use
Watch it while he draws out.
suction pretty
You talking
quickly
about
or I'll
that camera
surface
or
them.
me?
Apex.
The vehicle?
You're
Coming out.
in a good spot. Yeah, draw out.
Oops. Stand by. It's full out.
All right.
Go ahead, Bridge.
Looks like that.
Good copy. Thank you, Bridge.
Could retract that port bio pin when you
Yeah.
draw in.
Drawing in.
Yeah, stand by 1, Bridge.
Draw in.
And let me secure that pin.
All right. Push it back up, Levi.
All right, jar 1. Let's get jar 1 on monitor 12. And let's start inhaling, I guess. 500.
500 coming on.
Are they going to shoot backwards?
Yes,
Oh
probably.
yeah, they can swim pretty fast.
You want 500 now?
Yeah.
Coming up.
Left. Give me more left. Yep.
Probably should crank it at 1,000 and then
Okay,
back
give me
it down.
1,000.
1,000 coming up. What's 1,000?
Inhale 1,000.
Let me know if you want more.
You gotta try me those.
Gonna come back.
Increasing.
Yeah, just give me more section.
That's max on the joy box. I can give you a little extra if you need it.
Well, let me know what you see.
There he goes. He's above.
And of course,
Both-
they really
both of them
love
are still
hanging
there.
there. Actually, the smaller one would be, it's closer, would be the better one.
Got him.
Shoots in.
Yes.
Throttling down.
Is that the
Amazing.
only one you want?
If you see another one that is
Keep
is
trying.
catchable, sometime there was one
It's
kind
inside
of
the jar.
Copy.
He's on the run.
yeah. Then that would be great.
Increasing suction again. You got 1,000?
Yeah, he's wise to us.
Yeah.
What about the
A
one
little
outside
bit
the
bigger, a
coral?
little bit faster.
We're gonna abandon that one. What about the one on the left down here? That was in the base of that coral.
Stand by.
So come back.
You got a lot of dust in jar 1. Is that okay?
Is he still in there?
Yeah, he's still in
Do
there.
you see another option, or do you want to secure? Is there one in here somewhere?
There was one in that coral there.
Where is it?
Straight ahead.
I don't see it though.
I can zoom to it. Top edge of the screen.
Oh, there he is.
Oh, I see it.
See it?
Are you sucking?
Yep, 500. I'll increase.
More.
1,000.
Max.
Is he in? Did it go in, Roland?
He is in.
All right. Beautiful.
Yeah,
That's great.
Yep.
All right.
Suction
let's roll
Rotating
it.
jar.
Bring it back out. We'll secure all this. Boom.
Nice
Beautiful. Thanks for that.
Look at that teamwork.
job. All right.
That was-
Turning down. Stand by.
All right.
You probably won't want to pass back over that.
All right. You can draw her back out
Ready for drawer.
when you're ready.
I retracted pins.
Canister 1.
Coming out. Yep. Let me come out for it.
You're going to clean your hose, either suck or blow.
He'll blow it out under 6 in a minute.
Just remember not to cross back over 1 because they are swimming.
Yep.
Good job, Levi. Good job.
Beautiful
Good job.
selection. Thank you for that.
Chris over there.
All right, pilots, I want to make a heading change here. You guys go with that?
Yeah, so that's 3.5.
Pretty job now.
Suction
Hey, Rich,
canister 1.
pilot's ready for that heading change now. Thank you for your patience.
Chris, were you piloting or?
Suction jar light off.
Chris
Good
Wright
job.
on the sticks. Levi on the arm.
Taking my camera back.
And co-pilot on the hydraulics.
Got a 16-meter delta on you presently.
That was a classic teamwork situation. I've never been asked to sample one of those before. That was fun.
That's pretty
Oh,
neat.
good.
Got 2 of them.
The little one didn't stand a chance.
Pilot's first shrimp.
Yep. It tastes good tonight. All right, well, we're just going to keep going up here. I got a little bit of tether,
Oh, great.
a little bit of leash. Mudstones, huh? Mudstones.
Yes. Mostly
Mudstone massive. What's this red guy
Can
here?
we
Heading wandered pilot and coming back
zoom
to
in on that sponge real quick?
035.
Yeah.
Their snap. It just
Red
looks
is
a
a
little bit
mushroom
different
coral.
from the ones that we've been seeing.
Yep, there you can take this one. White color, yeah. Oh, interesting tubes.
Yeah, this one's super different from some of the other pharias that we've seen. Just a really different morphology.
Getting closer. I'm getting closer, and I'm touching down. Landed.
Wow.
Is that helpful, Roland, if I tell you that for
Yes.
focus?
Yeah.
Okay.
How much time do we have left on bottom?
Stand by 1, watch lead.
Thank you. Just as I think about
You're still moving.
collecting things.
I think we're all settled here.
We're just under 1 hour bottom time left. 5 minutes, approximately.
Nav, when did that last
Okay.
ship move complete?
That's great. I think we'll continue.
Roger, continuing.
We
See how
called
far
it
we
an
can
easy
get.
stop, so it's been a few minutes.
2 months, copy that.
Yeah.
Yeah. I think we're all good here. Settled up.
Copy. They're just making a heading change, so I'll give them a second. Go ahead, Rick.
You good,
Good copy.
John?
I think
Yeah,
I
we
see
can do
maybe
another.
a sea pen that's fallen down in the sediment straight ahead.
How do you feel about
Really big Brisingate up on the right.
035 again?
Yeah. Yeah, just stay normal to it.
Yeah, heading
Bail out right now, change.
Okay.
Is that what you're referring to, Bridge? Sorry.
I wanted to check out the
So you're thinking, I'm sorry, you said 2, what was it again? You were looking 225? Yeah, that should be absolutely fine.
You've got a little bit more leash here.
Roger,
Good
Bridge.
copy. 225.
Kind of
All
a
right,
sudden
you guys want 045?
truncation to these beds here.
Let's do 035 again.
035. Good
Thanks.
copy.
20
In
meters.
a line.
Bridge RV now.
Shrimps on the bottom right.
The
Hey,
slope
Bridge,
making
would like
me
to
think
put that move back in. We'll just do a
Directed to
full
the right,
20 meters
maybe
at
split away at some
035
point.
degrees, speed 0 knots to 2
okay.
knots.
I think we can keep on making our way up. We've got lots of
Okay.
zooms of these Brisingates.
They
Good
just
copy. Thank you, Bridge.
tractor me in. It's like a tractor beam.
Huh.
I get mesmerized. I'll keep
Ooh,
going.
we haven't seen one of those Solasters down to the bottom, right yet.
corner.
Copy. Sea star.
They're like sirens, hey?
Hello. Chris Ma here. Let's wait for it. All right, baby, take it.
Lights at 60.
Roger. I'm gonna get you closer.
So
Roland.
some kind of a a Solaster, I
Lights at
think.
60.
Well,
60 coming up.
Take it.
okay, now that we're closer, I'm less sure. And Chris Ma has kind of jumped in and out of the chat today, so if he doesn't see this now, I'm sure he'll really appreciate the zoom afterwards.
He's going with Solaster.
A Solaster, okay, great. It's a very beautiful pinkish color.
Got a lot of texture to it, a lot
I
of
think
spikiness.
there are probably quite a few different species, so maybe hard to differentiate in situ.
Nice.
Beautiful,
Full
thanks.
screen.
That's great.
Oh, another little shrimp.
And another really nice close-up parts
Lasers.
of these Actinarius.
They're out there.
Nobilis,
Get 'em down.
potentially anemones.
Clear.
Okay.
I think when we get extra time, it'd be
Lights
good to
coming
get super close to one of those shrimp so we can see the rostrum.
Okay.
Just so we've got it in the books for
Take it.
whoever's doing it.
Those larger ones might be
That's
a
not it.
little bit
That's a
different.
mysid.
Yeah. It's kind of hard to tell. Can we zoom in on the fuzzy one in the middle? Looks a little different from the other ones that we've been seeing.
Roger.
So not the one with the,
Right here. The one on
the
center.
snake star on it. Yeah, this one.
Okay, come in.
As close as you can get super small
Okay.
polyps.
Really small, whatever it is.
We'll get you in.
Huh.
On top of each of those white tips. Very
Yeah.
small projections.
Ooh.
Fog lights.
Fog lights
How much?
60.
60. On.
Come in. In.
Wow.
Centering.
What is this?
It's going to dust.
Chris Ma takes it all back. Cross aster.
wow.
Possibly cross aster.
Holding this
Ooh.
best I can.
I'm not
Paposis.
totally sure. Yeah, those do look like hydroids kind of colonizing this coral, maybe. I'm not sure what this one is.
It's
Yeah, like
Great
probably the
close-up.
stalk of a sea pen that's been colonized by hydroids. Very cool.
It's all that gold in the center.
Wow. Another one where the hydroids kind of took over and makes it look like a completely different kind of animal.
give a
That's
slow
really
pull out,
wild.
full reveal.
Yeah. Good job. You're going to get some dust on the bottom of it.
Okay, go towards the top. Try to get black in the back.
Roger. Towards the top.
Hold that.
Wow, very cool.
So does anyone know if these would likely be
Okay.
hydroids that are specifically
You're on the move.
colonized these
Okay.
old stocks and nowhere else
Thanks, Sean.
particularly?
Yeah.
It does seem
Yeah,
that
good
way.
check right
Like I haven't noticed any with
Good
that
enough.
morphology
Lights are clear.
anywhere
Coming off.
else along, the rocks and things that we've been seeing.
Fish.
Not the fish.
I kind
No,
of,
lights
I
back.
wasn't sure if it was
Snap it.
a sea
Clear
pen
pitch.
or. Oh,
Good
there we
copy,
go.
thank you, Rich.
Nice shot of these grenadiers. We haven't had one on this dive yet.
in.
All stockpiles.
Thanks.
So probably in the genus
We'll
Coryphaenoides.
settle pretty much right when we're at
It's
the
a few
shift.
different species, but these are the rat tail fish or
Shift
grenadiers.
change coming up.
Well, there go, video. Come out. I'm going to get off the ground here, out of the dust. Thanks, video. Proceeding uphill. Looks a little more sedimented.
Actually, can we snip a piece of one of those? Can I ask for a collection? Have that one
yeah.
straight ahead
Yeah.
in the middle.
Yep.
Yeah.
This one on the...
It's just, it's weird, and I haven't seen those particular hydroids do that thing.
Yep.
This is a great one if you could just use
Yep.
the cutters and snip off a piece.
Absolutely.
We can also see what kind of skeleton they're growing on. It looks like a dead sea pen.
Hydraulics are up.
Like you starboard. Starboard side's available. Right. Watch it. How much would you like of it? How many inches?
Go any light? Or are you good?
No, we're good.
That's probably enough, right?
Yep, that's plenty. If you can get it into the box.
It's a good snip. Shore coming out at your call.
Gonna give you fog lights. You can still camera entry real quick.
Lights coming on.
Alright, so we'll got.
Think we're good?
Alright, we'll start with it. I'll take off and we'll start on the starboard side.
Roger that.
I'm gonna take monitor 12
Go
for Sirius so I can see what I'm doing. Alright, here we go. I'm taking off. Auto heading and joy lock punched out. Taking off here.
We're settling up nicely on top of you here. Looking good.
Alright, you can probably
Yeah, every one of these sea
Draw
pens
out.
almost
Drawing
has a snake star. Their arms like dangling
Pull out.
down.
Alright.
You got your shilling cam over here as well?
Looking at
Nice.
7B.
Very nice.
It's going in.
Ooh, that's an excellent idea. Yes. ROV, if we could set up for a Niskin after this.
Roger that.
Thank you.
Ready? Coming in. There you go.
Starboard dinner.
It's full in. Full speed. And that's in. Retracting pin.
We're gonna do a pilot change up here.
Yeah, we'll do a
Sure,
pilot change and
sounds good.
pass on
Thanks
the
for
water
that.
sample requests as well.
That was sample ID EX2306-D15-07B in the starboard inboard biobox.
Okay, nav, ship stationary.
So last move was at 8 minutes ago, so it should all be stationary at this time.
Copy that.
Let's see. It looks like Sirius is starting to get a little above you, though.
All right.
Pilots, they were just about to get a water sample,
camera's
but you guys
5-0.
were ready.
Thank you, video. We got that in the pass down. How's it going, co-pilot?
Good. So you're down below me.
Yep, I'll push forward while you...
I'll let you get out of this dust
Okay.
before we take a sample.
Yeah, that's a good idea.
Camera's at 49. I'll tilt down.
We've had a correction that Sea Star we were looking at earlier is probably a cross-aster paposis, not a solaster as we originally thought.
Watch lead co-pilot.
Watch lead here.
Yeah, we were just trying to get a little bit out of the dust that came up there during that last sample. And we'll take the Niskin right now if you're still ready.
Yes, that'd be great. Thank
Roger
you.
that. Survey 2. Roger. Hydraulics are up. Reset. And here we go. 5, 4, 3, 2, 1. Niskin is closed.
ID EX2306
Okay, I'm
dash
pushing forward, co-pilot.
D15-08W. Niskin Bottle 3 at 2,041 meters depth.
Roger that, pilot. Nav, did you see that bottle?
Yeah.
So they've got a suggested ID
Same
from the
as
science
before.
chat. Those little hydroids could be Hydractinia, which is a group I'm not familiar with. Sounds very cool. Or at least in that family, the Hydractinidae.
Yeah, I think that happened before survey 2. It was a little crooked initially, and then it resets itself. And it was 3 that the same time.
I'm going to pull it up here real quick.
Yeah. Hey, Watchleads, this is Navigator. I have a question for samples. The last sample I have written down, the water sample we just took, would have been 07, but I think I heard 08. Could someone double check me, please? Sorbent import 07. Can you tell me what your 05 is?
Oh, I was wondering I was having issues maintaining my altitude, and I have auto depth engaged for the handoff. Much better.
Yeah, I think we're right on 08 on that last one.
I'm actually still looking. Let's see. So let's see. Your 05 is the starboard rock box. Is that correct? 05G. And then what's your 06? Canister 1. Ah, okay. I see it. 06B. Then 07 is the starboard inner we just took, and then 08 is the water. Okay. Thank you very much. Appreciate it.
So do we have when you're ready then, Nav, you should probably get a move in.
Yeah, copy that. We've been moving 035. You guys want another 20 meters of 035?
Sounds good to me.
Sounds good.
Really interesting lines in these rocks, too.
Mm-hmm. Little, I presume, bedding.
Ah.
Yeah, nice mudstone face, well bored.
I mean, all of this life seems to be really enjoying it.
It's hard to tell. It looks like it's lying in here pretty flat. It's not flanking the hills, the slopes like we saw in, I guess that was Nayas Canyon, but instead these are deposits that are once filled the canyon and are now being incised, and much younger than the conglomeratic and really hard sandstones we saw near the bottom of the slope, but maybe these are sitting on top of those rocks.
I'll center up then in a little bit, co-pilot.
Roger.
Now, in places like just near the bottom of the screen, those look like younger sediments. You can see but but more like mantling the current slope and not like these others that
What's
were
inside
actually
the
forming
cave?
pretty good
Yeah, that's what I
outcrops,
was
but
just
it's very
wondering,
flat
too.
lying. So, older and younger mudstones, we can think we can tell the difference anyway.
Can't really get super close.
I can try to zoom from here.
Yeah, I don't know if you have any light, but you can go in.
Never hurts to check, see if there's anything cool in there.
Yeah. No eyeballs looking back out at us.
Well, some little tiny ones.
Just a hole in the bottom of the sea. Full wide.
Thank you.
And white sponge may be of interest. Two different kinds.
Yeah, I think we've seen a couple of those at this point. Actually, I have a request to look at a black coral, but I think we passed it. So
To the right or below? Or?
Yeah, to the right. Oh, actually, that is a different sponge over there. Shouldn't have doubted you, Roland. Which one do I want to look at? The sponge that we were going towards, please. No, you have a great eye for this. It's yet another genus, I think, of glass sponge, different from the ones that we've been seeing so far.
Would you put the R3 on monitor?
Ooh.
What is that, 10 or 12?
Love seeing those anemones.
Thank you.
Laser is clear.
So this one looks a bit like a pinulasma often, especially from the inside or the oscular side.
Let's bring the lights up to 30%.
Often if we look at the outside of it, it has a bit of a different patterning, kind of more channels instead of big bumps, so it's possibly, well,
You can go in
I'm not going to
video
guess
when
until
you're,
we
when you're
get a
ready.
little bit closer. Beautiful, so you can see that really intricate, regular pattern that's caused by those 6-rayed spicules kind of interlocking and sometimes fusing together.
Do you want to look at any particular portion, Watchley?
I'll actually see part of the outside. So we're looking at like the inside of the hole, but if we could look at the side of it, which is kind of a tough angle from here.
Okay, I can move then.
Camera's looking 60.
Oh boy.
Oh wow, this is a beautiful shot of
Copy
this.
profile.
Like a mysid?
Yeah, full wide video.
You are full wide.
Wow.
All right, thank you.
Usually they're really small and they just kind of fly by, so. It's
Just
cool to
watch
get
your
that
swing
shot.
arms.
Nope, thank you. Should be okay, right?
Yeah. I don't know that you're
I can
going
watch it. I
to
think
get
it
the
looks
angle
good.
you want.
Yeah.
Your best chance is upper if you were to swing around to your upper right.
Yeah, I'll try and do that quick.
Just to confirm, and then I think we can add a whole other genus to the list of what we've seen on this dive. Yeah, I think this angle should get what I need.
Okay. Okay, video, you can go in.
Going in.
Great. Yeah, I think I'm seeing kind of folds and channels instead of those kind of bumps on Pinulasma. So this, I think, could be in the genus Homoiuridi, which is another one I've never had to say out loud before, just read it. But yeah, it's kind of distinct, easy to differentiate, or to mix up with Pinulasma. If you get a good look at the outside, like we have here, you can see these like folds instead of channels. That's great, thanks for that.
Thank you.
Camera down to 75.
Okay, I'll push forward.
Video's clear, lights are clear.
Some more of these really cool kind of folding tubes in the genus Pharia. Little mushroom corals, lots of that Actinarius anemone.
And pilots and watch leads, we have about 30 minutes left in today's dive.
That white thing upper right corner, is that a worm?
We're passing up through some of this more flat-lying mudstones.
See up below the pink mushroom?
No, I think we're good to continue. I think to rather cover a little bit of ground and just kind of document the bulk of what we see.
Understood. Okay. If you know where it was, okay, then yeah, I would just leave it out. It was definitely last shift.
It was the shrimp family.
I'm just joking with you now.
So ship stopped.
Okay.
Let's see.
I'm starting to get.
You're starting to get there.
Frontish.
Your blue view is pointed down for reference, so your blue view isn't
okay.
doing a whole lot at the moment.
I guess it's sample mode.
So we've come up about 200 meters from where we started this morning.
Drive it?
Yeah.
Yeah,
about 2/3 of the way up the side of the slope.
Right on the left.
It's another 100 meters or so to the top, perhaps a little more.
A different frequency.
Yeah, let's
we'll
switch.
not make that, but
all right, I'm going to take control.
Yep.
To the extent we can see what we can see on the way there.
It's one over. There you go.
And then
we'll see if we can cover
then
a
when you're
little
ready
bit of
now,
ground.
we could do that other move. I think I'm back out front again.
Copy that.
Is that a fish off screen left? Off screen?
Left?
Up.
It's 30 meters.
Thought I saw something swimming.
Yeah, I think it's a grenadier.
Pretty steep. Do you guys want to do just 10
We've
meters? Or
seen that guy before.
Yeah,
I think it'd be good for him to
I think that's probably
show us what we're
Albatrossia,
seeing
but,
here.
Copy that.
Yeah,
Or
we've
just
got some
our
really good imagery of that one.
angle. I think I might want to come to starboard some.
Yeah, your sonar's kind of showing that too.
Okay. That looks like.
Wow.
Would you agree, co-pilot? Looks like it's.
Huh.
That's kind of it right there.
Nice
Five-ish,
outcrop, nice
50,
exposure.
050.
That's cool.
I think
With
like
mudstones.
655. What's
060.
I wonder if
seems
these
to be-
aren't
Is that what we've been moving?
We've been moving 035.
deposits of glacial
But
flowers.
it's been changing a lot.
From
It was 90, then it was
glacial
60,
epochs
70.
that filled
So
this
it
channel
seems to be a little bit to starboard there.
during the glacier.
Okay.
During the the main part of the
I think that
glaciation.
pink stuff is different.
But then get mostly rinsed away during deglaciation when the bulk of the
Okay, so
sediments,
that's
especially
80.
the crust sediments, come pouring out.
Right, co-pilot?
Yeah. I think that's
Just a
just the
guess.
side of the mountain that you're seeing.
Would you call
Just
that
based on visual.
a glacial flower?
Yeah, yeah, maybe
Oh,
so.
I
Yeah.
like that.
A time to snap
Like
on
around
potential
there.
bubble.
I think we
Yeah,
go
that'd be great. I
060,
think it is a
20
bubblegum
meters.
coral.
Okay.
You
Works
guys
for
ready
me.
for 20?
Ready.
Copa, you ready?
Ready.
This could also be just a, it's so flat lying. Almost looks like what you would think that the deep sea abyssal plain sediments would look like. And that is something like that was certainly likely to have gotten caught up in between the collision as the Yakutat Terrane kept coming north, but it seems like it would be pretty strongly deformed by the time we got to it at this point here. So I think this is somewhat younger than that, more recent Pleistocene of age, something like that.
Since these look like layers on top of layers, did it like just settle and create layers like that?
Yeah, perhaps
And
seasonal.
How old might this be?
It could be anywhere from fairly recent to, if as I'm speculating, might be related to the glacial epochs as old as 2 or 3 million years.
Looks like you got some good stuff off left.
Okay. Oh, yeah.
Above, well, actually all directions, above, right, and left. Start to see it coming in your minute cam.
Could we get a snap zoom of that black coral? I'm not sure if it's the one that our science chat was looking for. Just straight ahead above the lasers.
Okay.
Just see if that's the one they were interested in. So there was one that looked a bit different from the one that we sampled.
And then here, interestingly, maybe the very same rocks now tilted 90 degrees on their sides.
Okay, you can go in video.
Laser's clear. Go to the area where the shrimp is. It's good there.
Looks like a similar shrimp, at least to the one that we sampled earlier. Seeing kind of a shorter rostrum.
And what's this transparent bubble like
like
thing
the
he's
slime.
messing with. Is that somebody's slime?
Yeah, it could be coming from the black coral, actually. They do create slime sometimes when you get them up in the sample container. Or it could be caught from something else that flew by in the water column. Yeah, just I think that's great. Little bit of detail for the folks in the science chat that were wondering, and maybe they can
up for full screen.
Type in exactly what it is that they're looking for. That's great, thank you.
You're welcome.
Video's clear. Lasers on.
Oh, there's a cool fish.
Where do you see it, Watchley?
Just up in the background there. Maybe a
Center
blob sculpin.
Up? Yeah.
Yeah.
I don't know if I can really sit down without wrecking a bunch of stuff.
Laser's clear.
Well, we'll just kind of get a snap as we go by.
Yeah. I think that would be good.
I'll try and I'll get closer and then we could go in.
So I have that, this is
Okay,
from
you can
farther south.
go in partial
Possible
video.
it's a different species, but I have the giant blob sculpin.
Oh, actually, if you come back out video, I
Cycroludes
cleared it.
frictus.
He's pretty blob-shaped. I
Yeah.
think that's an accurate description anyway.
Yeah.
It's a real good shot for the still camera too. Let's bring the lights up at about 30%. Yeah, just keep bringing it up till you see that shadow start to go away. That's good there.
Okay, you can go in when you're ready.
That's 60% rolling.
Thank you.
A lot of markings on the top of his head.
Yeah, I often see them like this. They almost look like scratch marks that have kind of, I don't know, healed.
Can't see
Lots
any
of little
scales.
spikes that look, I don't know, kind of soft, like they're sort of waving in the current. Beautiful eyes.
Got my focus.
I think they have, I don't know, it looks like almost like a little nose.
Looks very chill.
Yeah.
Video's clear.
Okay.
If you wanted, Pilot, you do have the tether to come around into that coral forest.
Okay.
Up to you, though.
That sounds like a good idea.
Wow, some
Lasers
really big
back on.
corals off to the left. Large stalked mushroom corals. Really densely branching black coral. I think I see 2 different species. Those really big bamboo corals and Paristonellas. A few dead bases of probably bamboo corals that have toppled over. Lots and lots of this Actinernus anemone. Sea stars. Wow, this place is covered. This is very cool.
fog's coming
Wow,
down.
certainly one
Yes,
of the more
please.
dense areas that we've seen on this dive. Whew, turning off the fog lights always makes it seem a lot more calm.
We'll just follow it a little bit farther
Interesting.
over, co-pilot, and go up.
Yep,
then.
you got the room. I'm up a little bit because the tether's right below me and we're taking some heaves, but you got plenty of tether.
Well, it's an interesting, this is an interesting kind of a bulbous nose that's kind of sticking out of the, we're going up a very subtle ridge on the way up the slope. This is an interesting outcrop, vertically jointed, but I think still are mudstones, and it looks to be a place where the corals are very happy.
Pretty nice heave.
Yeah, these,
Yeah.
I would say, are
Cereus
really
definitely
dense.
seems to be moving around.
Grouping of these bamboo corals especially, and quite large colonies. Interesting, they're all sort of facing more or less the same angle, at least the lower ones are all sort of facing the same way to take advantage of the current that's there. And interspersed between all of them is just also full of anemones and mushroom corals, different kinds of black corals, and a couple of primnoids, these really delicate branching white corals, but the bulk of these are bamboo corals, and really dense in this area. Could we zoom at the base there of this one? There's
The
something.
one with the lasers on it?
Yeah, something underneath of the mushroom corals. Can't really make
It's
out.
another anemone.
Oh, another anemone. Okay. Actually, we're good then.
Okay.
Yep. And then also several different species
Starting
of
to get
glass
out there.
sponges.
What do you co-pilot that? What do you think about?
Oh, can you turn? I think I
Which
see an
way?
octopus to the right. Right, that would help. It's a little octopus
yeah.
just under that bamboo coral. Always love a zoom. And it's quite a small one, so probably a juvenile, I would guess. They really actually blend in quite well with the anemones here. Kind of the same color palette. I almost didn't see it.
Looks like it's going to go for a stroll uphill.
Just having a stretch.
Laser's clear.
Yeah, just
There's
below I'm
really
seeing
a
like a
good
folded up
place
Actinernus
think I got
anemone.
one.
You can see it kind of opening a little
He's
bit.
waving his arms around too.
Yeah. And really easy in my mind to confuse,
Okay,
like
you
a
can go
closed up
partial.
anemone for that octopus, at least from the distance we were looking. Oh, beautiful. So I'm pretty sure this is a
Partial.
juvenile Granulodone boreopacifica. Reminds me of the ones that we were seeing in Noise Canyon, where we found those two octopus guarding or brooding over their eggs, and quite a few juveniles in the area.
All right, we have about 15 minutes left in today's dive.
Okay, can we watch this octopus for 15 minutes, please?
She sounds like a pilot.
Wow.
Does not seem to like touching that coral.
No, I wonder.
Okay, if you come out a little bit, video.
Yeah, I'm not seeing any of those sweeper tentacles or anything, but there could be a reason.
We can do another 20 meters. Sean.
Actually,
maybe at 6.5 this time.
Use octopus is the suggestion. Thanks, everybody, for the correction. It kind of looks like Granulodone, but I've been corrected. Muse octopus. So thanks for that. Really glad to have the science chat on standby for corrections in real time. I really appreciate it.
That's a good thing about our telepresence. It allows us to have the experts from around the world join the conversation.
Yeah, we definitely appreciate all the extra set of eyes. It's not always possible for me to flip through my ID guides.
Lights at 30.
The biserial suckers on the
lead towards the
arms
bottom.
and the smooth skin, I think, are both indications that it's a Muse octopus.
Oh, yeah.
So I believe that there are several species of Muse octopus.
That's good composition.
Not always easy to differentiate them, especially in situ, so I'm not sure that we'd get a species name, but possibly if we, with this really excellent video, we might be able to narrow it down. That's great. Thanks for that.
Lights are clear.
Can we pan over to the left and look at that sponge, the white one? It's also something different. Keep on seeing things that are different and new. Wow, this one's really weird.
Number of big blocky and randomly oriented blocks of these of this mudstone. And I'll say it again, this looks like things that were probably knocked loose during an earthquake and are somehow survived right on the edge of this ridge. It's a little less steep than elsewhere. It's not, I wouldn't think they'd come very far, more than maybe a meter or two.
Kind of hard to see any rock from this angle. There's a lot of biology.
Lasers clear.
Okay, you can go in video.
Thank you, co-pilot.
Well, a little more
hard
rolling.
to make out, but definitely another hexactinellid glass sponge. Kind of has a Faria-like skeleton, that thin single layer, but I'm not as confident about this one. So good to document. And with this level of zoom, we can do quite a bit. There's almost like a skeleton underneath. That's very cool. Actually, yeah, I've never seen one quite like this.
See how it's got like a stalk?
Yeah.
Almost.
That's a feature that I've never seen on any of these sponges from farther south.
Let's go back into the stalk on the lower left, right where it connects to the base.
Wow. Do we have time to possibly collect a piece of this one?
I'll have to move the ship back. Up to you guys.
Sure. If you pause
watch
it right
it's
now,
supposed to move the ship backwards. Is that of interest? If you'd like to sample.
Yeah.
Yeah, with our last 5 minutes.
Yeah, is that all right?
Sure. Yeah.
If it's
Beautiful
possible.
shot
This is
right
something
there.
that I've never seen
Great
before.
lighting.
Pi, it's that new finish. You're going
I've
to get
been
swing,
looking
but I'll
at
move
glass
you back
sponges
20 now.
for quite a
Copy.
while.
Okay, would you get a full wide thin video?
Full wide.
Thank you.
Lights coming down.
Is that too far away, co-pilot?
Yeah, you're pretty far away from there. Although your right toe is close. Yeah, that might be good.
Okay.
All right,
Is
Pi,
this
let's
gonna
watch
be a
swing
grab or a
coming up
suction?
here.
I think a grab. If you could just kind of snip some off of the end.
All right, co-pilot.
Yeah,
You're
I
going to be
think
my
a
eyes over
suction
there.
would
Or
just kind of
nav, you're going to be
give
my
us
eyes
a
on
whole bunch
12.
of broken pieces.
Put me up on 12.
On 12. Got it. Yeah. Understood. Your
Gotta help
camera's
be my
at
eyes.
7-0. Good copy.
Yep. Tilt you down.
Okay, craft coming.
Might want to come up a little, Bobby, too. You're, with the heave.
Yeah. I didn't want to pull them off with the heave, though.
Yeah, understood.
Looks like the sediment's
Coming up some.
continuing to blow worse.
That could be me. I think it's still enough I can do the sample. And it's starboard inner.
Correct. Starboard outer.
Yeah, thank you.
Panning left. Tilting up.
Thank you.
These are probably pretty kind of.
Did you get a little
soft
tighter
and
on the
fragile,
Zeus
kind of
video?
delicate.
That's
you can
good.
kinda
A little
get
back off a little
grab
bit.
of it. There should be a little bit.
And a little
that you
tighter
should work
on
with.
the mini Zeus, please.
Centering.
Copa, could you
Tilting
tilt your
down.
camera down a little?
Roger. Tilting down. Camera's at
Okay, what do you think about
That's good.
this watch, ladies?
Yeah,
Is that
that'd be
enough?
90.
great. Yep, that's
Copy.
enough.
tilting down. Oop. Down.
Okay, so I think we would have to come up and float, don't you?
Yep.
Wow, some really beautiful, huge mushroom corals, bamboo corals, and anemones as we kind of lift off a little bit.
Okay. Engage auto depth. Give it a moment to settle. And XY. Okay, XY doesn't seem as happy. I'm just going to come a little bit further off bottom.
You could also have your nav maintain your position.
Okay. Auto depth is engaged now. Just so you know.
Ready for draw?
Yep.
Okay, extending. Starboard should be open.
And would you rotate down a little bit for me, Nav?
Down?
Thank you. Yeah.
Copy.
It's fully extended. Following you.
Still not in the box yet.
Good view on Ox cam. Looks like you're down in there.
Okay.
You can bring it up on your left.
Okay, you're clear to close.
Closing. Putting up your main HD again.
And I just have a quick request. There are a couple loose rocks where we picked that up from. Do we have time to try to grab one rock? These might be slightly more lithified than we've seen, judging by the fact that these boulders are sitting here.
I think we can get
Don't
a
forget
rock.
to drop a point, Nev.
And maybe the one lower left corner.
I see it, type it out.
Roger.
Which rock are
I
you
see
after?
one lowermost left corner looks like it might be loose and
Get your box back on.
available.
I think boxes are back. You want to drive for it? Oh, no, you got it. I'll just keep the arm since we just have a few minutes.
Copy that. Where is it at?
Lower left corner, just barely out of the screen, kind of a flattish oblong. Yeah, there's a group of 3 or 4 of them, but maybe the topmost one might be grabbable, easily grabbable. Maybe we'll hold together with a... If it doesn't, that's fine, but just thought this might be a little harder than before, so we'll give it a shot if we have time.
Okay, I'll hold here till you're ready, Nav.
Okay, you want to try that?
Okay.
Which box is it going to be? Is it going to be port?
Stand by.
I believe port is available.
We may need to open the cover.
Yeah,
pilot.
you can open the box.
Okay. All right, you want to come in, board?
Yep, coming in.
Well, actually, let me tap it real quick.
Tapping, Roger.
Yeah, looks... I saw it move.
Yeah,
coming in.
Copa, can you rotate your camera up a little bit? You're starting to...
Roger. Rotating up. Thank you, Nev. That's 75.
Copy. Looking good.
Yep, position looks good.
Okay, you can go outboard for a moment.
I'll go out a little bit, but your cover might close.
All right, well, I can grab this one then.
Roger.
That was a different one, maybe, but... This one might do the same thing, and yeah, it looks like it did. All right, no worries.
Okay, sorry, Watchley.
Mudstones are mudstones.
All right, pilots, it is time to get off bottom.
Excellent.
Okay.
So the last sample was, and we'll likely get a Niskin here before we head up, but that last sample was ID EX2306 -D15-09B and the starboard outboard bio box.
Coming over to follow you.
Okay, that looks good.
Centering up.
Already
Okay.
watched lead.
Go ahead, Watchley.
When whenever it's a good time, maybe after we get away from the sediment plume up off the bottom a little bit, we'd like to request a Niskin.
Understood.
Thank you.
Sorry, Nav.
You're good.
Okay, I'm coming up off the bottom.
Okay, I'm going to start putting your swing arms away.
And I'll push forward out of this plume.
So those are really interesting dives. There was a lot of different things going on.
And I'd say whenever everybody's ready for the Niskin.
Nav is ready.
Roger, I got readies all around. Here comes the Niskin. Resetting. And firing. 3, 2, 1. Good fire.
Okay, looks like you're pretty much already lined up
I'm
with
going
the
to bring
ship.
your upper swing arms in.
Copy.
ID EX2306-D15-10W, Niskin Bottle 4 at 2,007
Get a little more
meters
altitude
down.
and line up with you, co-pilot.
Roger.
So yeah, lots to see in the biology of this area today. Kind of started off, and now we're back into an area full of these flytrap anemones, but eventually we got into, really diverse communities of different kinds of black corals, and then eventually made our way up to this area that we've been in for a little while that's just full of really large branched bamboo corals and some primnoids and still more black corals, lots and lots of these anemones.
Okay.
Kind of starting to get into a more diverse glass sponge habitat at the end here. So this is...
So you're already lined up. So then I need to turn a port to port.
And from the geology side, maybe everything I advertised. Here we are, a very complex area, and we did see a real mixed bag.
Well, I guess, so I would drive beneath you.
I didn't expect
to see, forward, and I'd be.
But yeah,
swing around
here
there.
we are in the Aleutian Subduction Zone, and actually the accretion zone, where the Yakutat Terrane is accreting to North America in this collision.
So what do you think?
As such, we might have expected to see.
So we're pretty well lined up.
Any number of different kinds of rocks in here.
I would have us create a delta.
At the bottom of this traverse,
this case
we
would
saw
be me coming up, like I'm doing.
Well-cemented
get a little bit of a
conglomerates
delta,
and
then you can back up.
Cross-bedded
I'll bring my camera
sandstones.
down when we're about 90 degrees. You can make your turn and push out.
Port turn and then push beneath you.
Channels.
Yes.
Go ahead. Deposits.
Go ahead.
I was just trying to think of a way.
Probably older age.
Be opposite of the ship then.
Older than 5 or 10 million.
Who will be
maybe
opposite
as
the
old as in the 30 to 40 or 50 million year old range.
We'll be set up.
Yeah.
All right,
So
I'm coming back.
But we know something
So we
like
go
that
about
is down
15.
here.
I'm
And
going
then
to keep coming up. I'm aiming for
sitting
about
on top of
19
that,
delta
some
as we do this.
more or less flat-lying,
My
extremely
camera
fine-grained mudstone.
is 90. So we can do this slow so that our tether has time
or
to
an interesting data point for people
match
doing
up.
more work in this area.
All right, so I'm pretty much right beneath you now. I can do
that's
the
why we
port
come
turn.
here. We never quite know what we're going to see.
Can take it nice and
This
slow.
is an area that's
Looks like
actually
the
very
tether's
hard to
going
study
off to my
by
starboard.
It's going
methods
forward and then
geophysicists
back over
usually
there.
use
So port turns good.
on the seafloor with sonar.
My camera's all the way
Sub-bottom
back.
information would be great,
So some of your momentum
these
was carrying you
canyons that we're
backwards.
in, there's a whole system of canyon channels that come down and are
I'm
meeting
going to
in this
turn
area.
to left
And that
just 10
becomes,
degrees so that I'm in
with
line
all
with the
those
ship.
different surfaces, a really hard
You're
place
aiming
to
for
image the
inverse of the ship.
sub-bottom with sonar.
Yeah,
I'm there,
So-
and now I'm going to push forward
All the
now.
way back. We got tether way up there. I'll bring it up on 5 as well.
The limitations of those kinds of traditional sampling methods, trawling and long lining.
Bringing up your oxalate
the ROV down there
for this.
Exactly the kind
As you
of
can see, you're right in the middle on down cam.
Yep, I see myself.
Yep, so about the time you come out of that view, you'll show up on the far right tether cam.
And I'll start surfacing
really
then.
great to
Yeah.
fill in that data gap for both of us, I think.
So yeah, not a lot in the way of geology samples, but pretty good job. Another banner day, I think, for biology. So
Yes, quite a lot of biological samples, and
Yeah, you can start to
every now
close
and
in
then
your
get a little
delta.
bit of geology
Okay.
in with them.
We're at
Not
20
sure
and
that's
you're
okay.
good.
maybe not.
Yeah,
No.
that
see
much
my
on
light
Which is
the
good,
sample
yep.
sample.
Light pool
Right.
there.
We'll see if anything's left in the
Yeah.
rock
See
box when it gets
visual
when
and
we get to
tether.
the surface.
I'll come out of
Yeah.
auto head,
Could be.
which will help us.
Well, I'd definitely like to take a minute to thank the
Yeah, there's one
GFOE,
football. Just
ROV,
waiting for our
and
second
video
one.
team.
We bring
You've all
oxalate
done
down.
an amazing job today. We're very excited with the video we were able to get in the
All right,
samples.
well, it's looking
And
better.
it looks good from both ends, so I'm coming out of auto head,
Also to
Okay, I'll do the same.
The crew
which
that's
means
gonna
things will
be
naturally
working in
follow the
lab
tether.
as soon as this ROV comes up to
And I would
sort
close in your
and,
delta.
prepare
Okay.
all of the
That'll help
samples.
get
We
rid of
really
those
appreciate your help.
up and downs on the tether we're seeing right
I
now.
also
All
want
right.
to thank the
I'm going up.
shore-based scientists, everyone that's joined us in the science chat today.
Turns to
We
zero.
always appreciate your
We're
time.
looking good.
Okay.
Bringing my
and
camera
your
up.
observations and expertise. So thank you for joining. Yeah, thanks to
So I'm
the,
gonna start to come up.
NOAA Ocean Exploration
Okay.
and the Okeanos
Ramping up to
Explorer
30.
for being
Hauling
the
in.
platform and the organization that made this all possible.
Cable looks good, or tether looks good rather.
Um,
Increasing speed.
Tomorrow's dive, I think, is what do we know about it?
It's planned for somewhere back in Prince William Sound,
Right. Yes.
as we avoid more bad weather.
27.
Stay tuned for details.
Okay. Keep an
And
eye
30.
on
I'm
the,
roughly on
the
30.
emails for shore-based scientists and the social media. We should be updating that.
So I
as,
think
as
maintaining a
things
little bit
progress.
lower delta and putting just a little forward
Thanks
way out.
also, Jamie.
Okay.
I
Kinda
really
keep our
appreciate
tether
all
happy.
Your observations and the geological context.
All right, I think
really
I got
benefit
it
from
now.
those.
And likewise, always great working next to you, Merlin. So thanks again.
Anytime, even tomorrow.
Okay, till tomorrow.
Okay, thanks.
You know, another thing we see that's such a joy on these dives is the fantastic camera work, the zooms, and just the detail of what we get to see at. It's a great way to see the animals in their natural state, as well as the geology when we otherwise can't really bring a sample back up. And I know Merlin can tell you how valuable it is to have these close-ups of the creatures that even if we can bring back samples of them, don't quite look as great when they get up here in the lab as they do down in life position.
Yeah, it's actually very important, super valuable to have that information, the imagery along with the sample. Often, yeah, like you mentioned, things look very different in the lab. It's hard to correlate between something that you see in a jar and what's actually living down there. But as we move to more of these image-based ways of surveying, kind of the less invasive ways of doing so, we need to have really good imagery of the reference samples that we take. And I've never seen a zoom like the one that D2 has. You can count, you can basically visualize individual sclerites on corals, sometimes spicules on sponges, things that often you need a good dissecting scope to see. So it's been
All right, I'm
fantastic,
gonna.
getting
I
all
think
of
I got
these
this locked in
really
on the zip
detailed
tie. I'm gonna
zoomed in
stow the
images
Orion a
of,
little bit.
diagnostic
Okay.
features of these
I
animals.
left that undone.
Yeah, but right now the ship is pointed into decimal 5
Video
knots of
crew is
current.
doing a great job. We
As
really
far as
appreciate it.
subsea current goes, looks insignificant
really
until about
highlights.
500, in which it starts to just build to surface current.
For science are often
Roger.
even more
Copy that.
interesting. You know, the really subtle, differences between sponge spicules. Probably not going to make the highlight reel, but it's truly important to me.
For the sponge dudes out there, sure.
Yeah. All my sponge heads. I don't know. There's maybe 2 of us. Great. So thanks for everything.
All right, we are elbow up, wrist right, shoulder left. Well, let's start with a little bit shoulder right. Shoulder right. Here's some shoulder right. Now we go shoulder left, all the way out. Shoulder up. Elbow down. Elbow down and wrist left.
Actually, do we have a Niskin on the way up at all? Got it. Thank you.
Disable valve packs. And we're good. Hydraulics coming down.
Copy.
Hydraulics off. Still spinning 30 meters a minute. Anything?
Let's see. Empire's update from Dive Super. Around 1500, so if I zoom out, this is about 1800 here, so we shouldn't see anything past that. So once we get to around 1500, Dive Super would like us to start get out of autos. Decimal 5 through the water right now is pretty much just going to point us right here, so it might have us come a little quicker. Maybe like decimal 7.
Okay.
Or if streaming's fine, we could just hold this. But he wants us to get out of auto so they can play with heading. I think it's going to be a tricky recovery.
At 1500?
At 1500 per Dive Super's request. So if we do decimal 5, we'll just hold station. Does that make sense?
Because we'll be 300 meters above the tallest bathy.
Correct, and we should just hold station in addition to that, as far as bathy goes. Comments, questions, concerns?
I have concerns of coming out of auto sway that early.
Okay. We can leave it in. We're pointed directly into the current, so auto sway shouldn't affect us that much.
Roger.
But I recognize your concern, and I see you.
My concern is coming out of autos that early when we're still way down, because we're not going to really be seeing anything down there.
That is
so
what
if the ship
ACP
starts
is.
going anywhere because of high currents, like
It could swing Sirius around.
Which isn't.
I could leave autos in if pilots are uncomfortable removing them.
No, I like your idea of coming a little bit faster to guarantee that we're making forward motion over ground. Because if we just do half a knot, and then that ends up moving the ship backwards over ground.
Yeah.
Then we get the vehicles underneath, and still not a problem, but just not a comfortable position.
Agreed.
So I like the 3/4 of a knot forward. I would probably keep auto sway in, but if Dive Soup wants it out because they're going to be making heading changes, that's fine.
I could leave it in, and then if I see him, I could let him know, or I could just let Bridge know we'd like to, unless they need it for heading maneuvers, we'd like to leave it in.
Yeah, I mean, if they're doing a big heading,
know.
as soon as they come about anywhere, they're going to have to come out auto sway.
Yeah, but they could let us know when they're doing it.
All sounds good. I like your plan.
Copy.
Stick to your original plan.
ROV watch lead.
Go ahead, watch lead.
Yeah, we'll get a last Niskin at 350 meters.
350, heard.
Will do.
Thank you.
And yeah, Bob, I think as far as that one Niskin goes, like, the design of the Niskin heads, like, they don't have to be upright to seal.
Yeah.
Pretty much any angle they're going to seal. I wonder if that one seals a little more crooked is because the tension, that hose might be a little tighter. So it's mating the first edge it gets, but it should still seal.
Yeah.
That's my working
Could
theory.
also be under less tension because it's the middle on the release, right?
It's true.
Like it's more lined up with the release mechanism.
It is more lined up with the release mechanism.
Where the other ones have to pull further to get into the release mechanism.
Yeah, but that specific hose was the one I just replaced. It's
True.
definitely the most tensiony of the...
Yeah.
Yeah, I just noticed that 3 pop back as well.
Yeah, eventually it works itself centered.
Yeah.
But I think as long as they're popped, they should seal no matter what.
Yeah.
Yeah, the wind was supposed to die down throughout the day, but instead it just built up and came at a more awkward angle. So the dominant swells are coming out of the east around 090, and we're pointed into the current, and then winds almost opposite the dominant swell. So right now we're kind of splitting everything, but as you can tell, it's going to be a little bit of a bumpy ride.
Raj.
Raj.
Starting to swing around a fair amount here.
Yeah, I just put on a little bit more forward.
Let me check our, let's see, 1650. All right, I think we're right on time, gentlemen. The current's actually clocking a little bit, which makes us... Do you know approximate heading that you're interested in? Is this like a 5-10 degree move, or are we talking 50 degrees? Just trying to get an idea. Pilots 2 copy.
Yeah.
Copy.
Increments of 10 to 3. Are you comfortable with that at this time? should help with going into the wind, so should help with streaming a little bit.
So as they do this, they're going to come about into that current, which is what, half a knot?
Yeah, in which case I definitely don't want to remove auto sway.
Yeah, so we need to keep an eye on that.
Yeah, because even now,
because if they come around 30 or more, they might have to punch out.
Correct.
Especially with the thruster, stern thruster that's not behaving.
Hey, Rich, yeah, we're ready for that. If you could just, if you hear the thrusters ramp up and come out of auto sway, yeah, please let us know ahead of time. Okay, understood. We're ready.
Are they still A-frame?
Hey, Bridge, over that. Hey, the center rotation's still A-frame? Or no, it is back to midship, correct? We got it.
Yeah, 'cause that would swing you closer to me.
Yeah, they put it back 10 minutes ago.
It's okay if it's midship. I was curious.
It would just be hard, yeah.
Your forward way is enough to not worry about me swinging.
And pilots, when they're done with that, I'd like to start getting us moving ahead. What do you think? Keep auto sway in, start coming ahead. 0.7 3/4.
Yeah.
Sound good?
Sounds good.
Copy that.
And that was 1,500 because the deepest was 1,800.
Yeah, now we are
Or a little
pointing
shallow.
more. Obviously up now. So now this is like 1,700 here.
Okay.
But we shouldn't get anywhere close to that by the time
Okay.
we're there.
So it does look like you have enough forward way on pilot that you're actually starting to pull me.
Okay.
You might want to back off a little.
All right.
Pilots ready for me to start having the ship come out auto surge and make forward way?
Did they say they're staying at this heading?
They said they're done with their heading change.
Yeah. Good. Those are so cool.
But now that we're pointed not in line with the current. So 0.7 might be kind of hot now for you. I might watch his average for a minute before I call in a move.
Okay. So you're when you were saying I was pulling, you're talking about Sirius moving to the east, right?
Yeah.
Well, and D2 can do that? I thought it would have swung a little to the east from the heading change.
It would a little bit.
D2 can pull.
But it also kind of seemed like your trend was starting to pull me over.
Okay.
Which isn't necessarily bad. It's just better to kind of get an equilibrium where you're not causing me to move, creating layback.
Pilots, I might start with like 0.6 through the water. You okay with that? I think that it's dropped a little bit.
Okay. Roger.
All right, put it in. This time, if you're comfortable, we'd like to come out of auto surge and start making a 0.6 knots through the water. Good copy, thank you.
Put more on because I kept on seeing the slack in the tether, and didn't want it to snap. Wasn't sure of any other way to prevent that.
Yeah, but with this heading, I'll probably just leave auto surge all the way up until 50, or sway, because I think we'll just start screaming west otherwise.
Okay. Yeah, looks like the surface current. And that would be the surface current primarily doing that, right?
Yeah, that's the surface current would do. And we're already trending this way. So the ship would do that. The vehicles would just trail. I don't think it would be good for streaming purposes.
Delta's getting a touch low.
Okay, I backed off a little while earlier. I'll do some more.
looks like our forward weight dropped a little bit. Can we increase one more a little bit? Okay, copy that. Oh, what was DVL today? Looked like it wasn't great. Did anyone watch it closely?
I didn't. I tried to use XY during that one sample.
It was really silty today.
Mm-hmm.
Wonder if that might have been anything.
I think it kicked Fernando out earlier during the sample as well.
It was pretty steep today, and I mean, it normally, when it's very steep, it normally doesn't work super great.
Yeah. Can I put up the aux2 camera on this monitor?
Sure.
Make sure the hose is still there. Oh yeah. All right, the DVL didn't fall off, so it's there.
For a second there, I thought it like was crooked, like something had hit it or something like that, because I saw the one beam, and I was used to looking at the tabs, and it's just one transducer. So what do you guys think? Should we buy a bunch on eBay, or should we try and get a demo for a Ranger 2?
I say both.
Is the Ranger 2 the one Levi's been looking at?
Yeah.
Yeah, let's do both.
It reports 25 hertz update rate, which is insane.
That's a lot of hertz.
Yeah, I mean, it's more than double the highest I've ever seen. By 1 hertz.
All right, so Niskin 350. Understood. Let's see, we only have one jar sample. Canister 1.
All the boxes are full. Just one can?
Just one can, number 1. I think that's a shrimpy one, so someone can bring that out.
And all 5 Niskins. By the time we recover.
Yeah.
So that's from heave, or should I push forward?
I think that's just a ton of heave. I don't know how much you can do about that.
So pushing forward, I feel, actually makes it worse because it keeps the tether under tension.
Okay.
Whereas if there's slack, you know, it kind of same thing as on bottom, right? The heave won't work its way all the way down to D2 to cause the tether to go tight.
Okay.
Right? It'll sort of just float out in the water.
Well, I think but if the headings get misaligned enough, and then you have like tension in the tether, for example, like because D2 is forward way on, you could snap, and I only know this because of experience, you could snap a tether. Dive super. decimal 6. You're doing like decimal 5. Or I can do the one that we have. Yeah. Wind died a little bit.
Yeah, this
Now
is
it's a little chunk.
so. Yeah, it's a challenging sea state that we're gonna have to deal with. This seems like the best heading so far. I don't know if you guys just felt it a few minutes ago. There was definitely a pretty
gnarly breeze,
significant roll. So that might have to be timed if things don't adjust. So I'm going to go spend quite a bit of time out there. But yeah, if you want to maintain sway engaged in your stream,
I'll probably leave it in unless we do that. Unless it's changed. I'll probably just leave it in if it's okay.
yeah, that's fine. I'm going to probably go get my headset, get ready early, and just stand out there. Make sure there's no static pickup.
Sounds good.
Great. Andy, a lot of focus on this recovery, boys.
A lot of energy.
A lot of energy, a lot of focus.
Yeah. All right. Let's keep our heads on a swivel.
Copy that.
Appreciate you. Nice work, Bobby, with the lever. Doing great.
I wanted that rock. I like the one where
And have the ship come forward a little more. So one reading I'm seeing about decimal 5, decimal 6, maybe like decimal 7 knots a little bit more. Oh yeah, Bobby. So what happened to me the one time was going in tow like this, and then there was like a heave or cross current, quickly turned Sirius heading 90, and I had forward weight and joy lock locked in, and it just snapped and broke the tether. I think that was a very abnormal event, but it did happen once to me.
Yeah, I remember that.
Wasn't a great day for me.
Didn't seem, I don't know, it didn't seem like anything that you did differently.
Bad timing, bad luck. Definitely wasn't a lack of skill.
If the workhorse starts misbehaving, I don't know what I'm going to do. Oh yeah, we could make up some data. It's our speciality.
It just takes Sirius altitude, subtracts delta.
There you go.
Add in a decimal point.
Yeah, that moves
Random.
around a little bit.
Yep.
I like it.
I heard you say that. We didn't have any serial data issues.
One nice thing on the Tasman, we're definitely not flapping in the water column. The same thing on the way down. Actually noticed that the DVL recording wasn't being made because it doesn't record anything if there is a bottom lock. And all throughout the cruise this far it had been flapping all the way down, so it was creating the file so the little alarm that it wasn't being written didn't go off.
So regarding Joybox, do we want to take this opportunity? I mean, we can talk about it more in the meeting, but do we want to take this opportunity to upgrade any of the smarts inside?
Yeah.
Perhaps the Pi?
Yeah, I think
we'll do the changes we already talked about, but just as far as, you know, these models are how many years old?
Yeah, I'd say replace them all.
Acquisition might be a little hard, but because I know Pi's are on back order, but it seems like we could upgrade to the latest model.
Yeah.
That doesn't require many changes on your side.
No, it wouldn't.
Okay. We'll need to do a size check. Hopefully the footprints haven't changed too much, because beyond what we have in spares, I don't even know if we can buy these models anymore.
Yep.
The Arduino, I think, but not the Pi.
I just looked. I was hoping this one was alpha, but it's spare. Alpha was the misbehavior last year.
Okay. Yeah, these are both old style. The one over there is Rev A.
Okay.
It did not have a alpha designator on it.
Okay.
I don't believe.
That was the original problem child, if I remember correctly.
Yeah, I think EE will do a better job of marking these as well. We should be on, well, we can discuss it later, but we'll get some labels on or something definitive to mark these as what they are.
Sounds good.
Their insides and, you know, maybe a serial number or something.
And I think Fernando already figured out a good way to make it easier to swap boxes. We just have to calibrate them all once, and then you could bring it in and you won't need to worry about the calibration. And the other thing we were thinking of is using one of the bulkheads and putting a thumb drive on it and logging the system logs from the Pi to that.
Yeah, if if you need additional connections, let us know.
Okay.
Stuff that, because right now I think it's just, what is that? Right now it's just the 3, I think. But if you wanted a keyboard or an additional one for USB stick logging or whatever, we can add those.
Yeah, I think the big one's the logging, because like whenever it locks up, we only ever notice. This one locked up in between the power ons and the launch.
Yeah.
After I had bounced it an hour earlier than that. But the Pi is definitely involved because I can't access it over the network whenever it locks up. But it's hard to troubleshoot.
We'll take a look at the connections then to the bulkheads and see if what we have is something you can. I don't know if you'd be able to plug into a USB drive for that, or if you need to be connected to like a different USB port or something.
Yeah, we could. If we just open it up, I think we could put one in there.
Well, it sounds like it'd be something good to have on the back for future.
It would,
for all these, I'm thinking future. What's on the wish list.
The other issue is I've never been able to make it lock up when it's doing something other than being a pilot box.
Something about the pilot software or routines.
It seems like it has to be. The software that, it's the only difference is the GUI. And it's the same binary, it's just a different configuration for it. Same code, I suppose. It's a Python one.
Do you guys track the Joybox maintenance anywhere? Or like Alpha, Bravo, Charlie? Do you track any of that beyond just a sticker on these?
No.
Okay.
We reference the sticker and like Trello cards, but that's it.
But otherwise, I think that's fine. I was just trying to think of
how to track them.
Yeah, my mind wandered off. Not so much the Joy boxes, but anything that we start to get a handful of. How do we remember what our last letter or serial number was? So that we don't accidentally end up with 2 foxtrots or something.
I think we just start fresh. We'll update some stuff over the season, and there'll be Bobby 1, Bobby 2, Bobby 3.
There we go. I'm trying to get a laser etcher for the shop, and then we can just engrave a serial number on the base of all these. Really? Do you have to mount it or something when? It comes with a magnet mount. Okay.
Free-handed?
Does it, as in it goes fast and quickly enough that your wobbling doesn't matter, or they just don't care if it? Okay. You could make a mount for it if you wanted a very clean, etch. That's pretty neat, because that's the problem with, I like that because, laser etchers, a lot of them have a maximum thickness. We'd never be able to fit for the price range we'd be able to get. You couldn't shove a hole. You'd have to put in the bottom plate when it was just the plate. Yeah. Well, that would be to make the whole panel ourselves, but if we wanted to start etching just label names or serial numbers, that I kind of like that. You have to show me this device. The idea of a portable one's super neat. I missed my 10-minute call out on Niskin, passing 600 meters. .350.
It's like a very short range lightsaber.
I want one.
Etch your name into somebody's
I was going to say, we're going to start ending up with...
Monitoring deck Sonar.
.500 meters, 5 minutes away from the Niskin. Unceremoniously. Oof, that was a rough set. button, button, e-stop, knife. Does the co-pilot's button turn everything off? Okay. That's what I heard. I know what we should make it do. Acoustic devices only. Boom. Off when we get like to surface or something. Or we maybe just leave it doing nothing. Could do acoustic and lights maybe. Or just acoustics for surface. Oh, for like turn on you mean? Yeah, could do it for turning on stuff, yeah. .380, 1 minute to Niskin.
Argonaut Bridge. Roger, we can disengage. Are we looking to come out of sway anytime soon? Roger, okay. I'll let you know if we start to lose heading at all with the sway engage.
Survey 2 and watch leads, 15 minutes to Niskin. Sorry, 15 meters to Niskin.
Copy that.
Five, 4, 3, 2, 1. Firing. Good fire.
Sample ID EX2306-D15-11W, Niskin Bottle 5. All right, thanks, gentlemen.
Thank you, watch lead. .300 meters wire out. You could, now that we're getting shallow. The ship's making 3/4 of a knot forward. Probably don't need to push out really. I'm not saying it's your fault, Nav, but other Navs don't get the NAs. Yeah, I mean, we're probably shallow enough that we're feeling it. I would take it out soon. Let's do it. Passing 250 wire out.
RV nav bridge. Roger, taking auto's way out, and we'll try to zero us out laterally here. RV nav bridge. So just for general awareness, it looks like the wind has clocked off to port again. We were looking to potentially point further into the wind for recovery. There's not a great heading that we can hold for a good ride, so I'm sure they can make adjustments back there, but yeah, just for general awareness, we may look to come more to port for recovery. Roger, yeah, that's what I was thinking. Yeah, they can make that decision back there, but I'll give them an update when they call for comms. Yep, no worries.
I agree with leaving it to Dive Super because he picked this heading specifically. It did.
RV nav bridge.
I think right now.
Roger, we are actually, yeah, we're getting really variable speed changes up here. We're going from 1 knot down to 0.4, so it's been alternating.
Okay, understood.
We're roughly holding 0.7, but yeah, as you can see, it's really fluctuating up and down right now. I think it's just the swell is taking us.
Okay, understood. I see us moving. Looks like just a little bump backwards over just for feet.
Roger, yeah, we can try to keep that average up higher, 0.8.
Yeah, that sounds good.
Okay, sounds good.
This shallow, we're probably seeing surface currents anyway, right? So even if we're going backwards over ground, we're still streaming nicely through water.
Dive Super, comms check.
Dive Super, this nav, loud and clear.
Loud and clear, Nav.
150 meters.
Pilot, Dive Super, comms check.
Pilot reads you loud and clear, Dive Super.
Loud and clear, pilot. Winch, Dive Super, comms check.
Dive Super winch, got your limit, Charlie.
Loud and clear, winch. Deck boss, Dive Super, comms check.
Loud and clear.
Loud and clear, deck boss. Bridge, Dive Super, comms check.
Loud and clear.
Loud and clear, bridge. Nav, Dive Super.
Dive Super, go.
Can I get an update from your perspective, please?
Yes, sir. Ship's pointed at about 10 degrees, so we're splitting the surface current, which is 0.7 knots right now. That's off the about 30 degrees starboard up the bow. And then the wind's on the opposite side. We're seeing about 15 knots right now. It's clocking a little more, so it's maybe 60 degrees port side of the bow. Auto surge is disabled. We disengage auto sway about 200 meters to help with streaming. Streaming's looking pretty good right now. Maybe, let's see, 20 degrees port, but that should improve with auto sway out. Port prop and rudder are disabled. Center rotation's midship. Auto surge is also disabled. We're a little hot through the water at the moment, but you could probably bring that down as soon as you guys get control. I think vehicles are pretty happy at the moment.
Copy all, Nav. Are you ready to pass comms to Dive Super?
Yes, sir. Dive Super's comms.
Dive Super has comms. Bridge, Dive Super.
Dive Super Bridge.
Can I get an update from your perspective, please?
Yeah, so, I can confirm with the RV nav, the wind has clocked off to port a whole lot, actually. So we're no longer pointed bow into the wind. Seas are still looking pretty confused. I'd probably recommend coming off more to port just to get your bow further into the wind. We don't have any traffic in the area. I've been fluctuating
7.5 meters wire out.
So it's probably going to be pretty variable. You're just going to have to make a bunch of adjustments to maintain speed through the waters as desired. Aside from that, though, no new updates.
Copy all, Bridge. And can you confirm center rotation is midships and port prop and rudder are disengaged at this time?
Roger, yep. Port prop and port rudder are disengaged and center rotation is midships.
Thank you, Bridge. I'll have you work with AFCON to get control pass back here.
Roger.
.55, winch is slowing.
Copy.
Dive Super.
And holding 50.
Dive Super, go.
Just for your awareness, we are passing control to the aft station at this time. Please watch streaming as we do so. We're going to adjust that forward speed down to decimal 5 and try and zero out the laterals.
Good copy. Vehicles are streaming center line right now. Looks good.
Good copy.
Dive Super Pilot, vehicles are holding at 50 meters.
Good copy, pilot. Holding at 50.
Tucking all hydraulics.
Winch, Dive Super.
Go, Dive Super.
Can we pass control to the aft station at this time, please?
Aft station has control.
Look at that jelly. That's cool.
Aft station confirms control of the winch.
Okay, I'm pushing out. There we go. Co-pilot, you seem to be moving up and down vertically a little bit.
I'll work on that.
So when we have the big keaves, it goes up and down almost the whole view of the tail cam, and then goes off to Sirius's port. So there isn't really anything I can do about that except for push out.
The headings?
Yeah.
Yeah, I think just add in a little bit of forward. Lock it in. They're trying to find their happy spot as well. You know, you can see these S turns means their percentages aren't dialed in yet.
Bridge, dive super.
Dive super bridge.
Do we have permission to continue with the recovery?
Yes, you do.
Thank you.
Yep, nice.
Control room, or pilot, dive super.
Go ahead, dive super.
Is the control room ready to continue with the recovery?
Control room's ready.
Okay, pilot, let's go ahead and ascend up to surface. 15 meters per minute, calling out every 10, which will do the same.
Roger.
Ascending.
Dive super for reference, vehicles are streaming about 20 degrees starboard at the moment.
Copy that, 20 to starboard. Four 0 meters, wire out.
D2 crossing 40 meters depth.
Three 0 meters, wire out.
D2 crossing 30 meters depth.
20 meters, wire out.
D2 crossing 20 meters depth.
Good visual of the ship.
10 meters, wire out.
Video, would you swap it? D2 crossing 10 meters depth.
Visual agrees with high pack, off to starboard.
Right, pilot, we have eyes on D2, slightly off to starboard. We'll bring it center line.
Hose on A-frame. You're just past it. Yep, right at the hose on the A-frame.
Roger.
Right above the crane
That's
station. Right above the bouncer ball on you.
Okay, pilots on surface, go ahead and lock in auto head in versus ship.
Auto head engaged.
We're walking you over.
Giving it some bumps to the surface.
Copy that, pilot. You might have to give quite a few today. You're going to be hit under from all the waves.
Understood.
All right, deck, let's go ahead and recover Sirius. All right, pilot, if you could give some halfway and reduce the tension of the tether.
Backing. at 60%.
Good copy, pilot. Hold that. It's good, pilot. Let's maintain. Maintaining, pilot.
Maintaining. Should I increase dive suit? I'm at 60 still.
Go ahead and increase.
Okay. That's 80.
Pilot, we're using our four-way slightly.
Copy.
Winch, take back local control.
Remote disabled. Sirius is clear.
And pilot off stick.
Pilot off stick.
Let's go ahead and leave auto heading engaged until about 2/3 of the way down the tether.
Copy. Auto heading disengaged.
All right, pilot, go ahead and disengage.
Auto heading disengaged. 0 thrust.
Good copy. Lift lines clear, daisy chain free. Okay, pilot, lift line is tight. Let's go ahead and lock in auto heading, inverse the ship, and start backing towards the crane.
Auto heading engaged and backing.
You could give us a bump to surface, please, pilot.
Coming to surface.
Another bump to surface, please, pilot.
Coming to surface.
Nice work, pilot. Let's maintain that nice and easy back.
Maintaining.
Okay, pilot, let's go ahead and ease on your back.
Easy.
It's good there. All right, go ahead and increase.
Increasing.
Looking good. Keep backing, pilot, looks good.
Backing.
And a small twist to starboard, pilot.
Twist. Auto heading
And
secure.
pilot off stick.
Pilot off stick.
Your vert's faulted there at the end.
Yeah, I just saw that.
You want topside power.
Pilot, secure power.
Deck E-stop. Winch. Knife switch.
Knife switch.
0 voltage.
Vehicle's cold.